It's about the time start this event. Actually, I'm really appreciated that some of the all invite speaker comes from some of the very different places, right? And that is from two professors from America and two professors from three professors, right? And from Japan, and then one researcher in the Sumitomo pharmaceutical, like four of them from Japan, right? And then one is Australia. Two, yes. Two is Australia, and then another one is from Germans, right? I think that's what we invited, and then it's now it's Peter Seeberger. He just landed down into some Incheon, and then now he's coming too. So that's why I arranged his talk on the last part, right? So actually, this is for the we call talk concert, like not really like a music concert, right? It's the talk concert, and then this is for the public, and then it's some as you see. And now it's also people in the busy, and then it's only few some of the attendance, and then coming over here, unfortunately. And there is please understand it. This is a kind of some of the bureaucratic event, right? And then also maybe please try to understand it. This is a kind of some of the relaxation, the event, and that is some of the opening event, right? So then tomorrow we can move to the Busan, and then is some of the only invited, attended, right? Working in the industry, in the academic, and there is a research centers, right? So that is some of the 2 nights, 3 days, and there is these 40 people, and then like a golden conference, and then we are going to gathering together, and they stay in the same hotel, and then we're going to have a dinner, lunch together. Then it's quite closed, right? And then so I'm really expecting that some of the event in the Busan, there will be the really real core, like some event of these symposiums, right? So actually, I really like to appreciate it for some of the sponsor of some of the KOFST, and then Korean Federation of Science and Technology Communities, right? That is the KOFST, right? This is kind of a semi-government, like some of the organizations, right? It's mostly like some of the finance is supported by the government, like a budget, but then there is also supporting some all kind of academic activities in Korean, the research communities, right? So it's whatever biochip or flow chemistry or chemical engineering, mechanical engineering, all kind of some academic communities belongs to some of the KOFST, right? So then, and there is a KOFST are going to be supporting, and there is a kind of this kind of some international global conference, and there is some that or it's a kind of some the controlling tower, or convergence of some opinions in the scientific community to try to communicate it between some of the academic people and the government peoples, right? So that's why, as you see over here, and that is located at the core of Korea. So you know, 그 가수 이름이 뭐지? Psy? Yeah, Psy. You know the Gangnam Styles, right? Yeah. Gangnam means that is the place over here. Name of a place over here is Gangnam. So probably you get off the Gangnam station, the subway stations, right? So and that is unfortunately I cannot dance in the Gangnam station. Maybe if I take it as quite heavy drunken, and then in a little bit psyche like a sound, and that is probably I can make a dance. So yeah, I think that is one of the richest area in the Seoul. So there is some that kind of like a Korean Wall Street, and there is some nearby over here. So I think that hopefully you may have a time look around, and that is in the evening or in the night, 2 nights, right? So this is a really good place and to stay, and that is. But I think that every metro city, every country, almost no difference, right? So, but if you come to some over the river and the northern part that is more traditional. So, and that is after this conference, and maybe Mike, and you have a plan to look around over here. So, and when you have a free time after this event, and then I really enjoyed some of your free time at here, right? Okay, so appreciated some of again and to some of this cost, and that is also appreciated some of the all attended the audience and to these symposiums, right? So that's why some of I assigned only the 4 speakers, right? And the 4 speakers is some of don't be make it too like professional was specials, right? Make it very general talk, make it some of like quite relaxed, and then we make it some of the more easy communications among the people over here, right? So it kind of we can practice, right? And that is that is a real core event, and it's held it in the Busan areas. So Busan is also our hotel is located at some of the like a coast area, so there will be the really another. I mean, we will touch another atmospheres, right, in Korea. Okay, so that's all, right? And maybe I think that in the event, and then we're gonna take it to some of the like a video, right? To watch the video. Any? Yeah, it's a video. It's very luxurious, right? Okay. Actually, let's start it some of our main event, and then is as I told you, so we have two speaker at the first, then and then after coffee break, and then we have another two speakers, right? So and each speaker has some 40 minutes, including the Q&A times, and then so. Okay, so first speaker, and then let me introduce is Professor Daeyeon Lee. He's from the University of Pennsylvania in USA. So actually, also is personally I'm interested about your work because we are working toward quite similar areas, right? So and then it's hopefully, and our group member and then as other member really enjoy this talk. Okay, please welcome his talk. Mm-hmm.
Okay, well, thank you very much, Professor Kim. It's always a great honor whenever you call me to visit Korea to participate in events. So my name is Daeyeon Lee. I'll be telling you about how one may use, what I'll be telling you is how one may use microfluidic platform to accelerate discovery and manufacturing in biopharmaceuticals. I was told that this was going to be for general public, so I left out a lot of the technical details, and I was thinking of my son, who's sophomore in high school. So please do understand if you see materials that are look very basic. So let's first start by looking at what biopharmaceuticals are. Okay, so these are, of course, pharmaceuticals, but biomacromolecules that are from biological sources, usually living organisms like the cell or plants, and even enzymatic reactions. And these types of biopharmaceutical, market is growing quite rapidly. So you see here, right now, it's around, $500 billion, but you can see, in 10 years or so, this is going to be a huge market. So there are a number of companies in Korea that have started a new division or a completely new entities to sort of catch up with this trend in biopharmaceutics. So the thing with biopharmaceutical, and this is not just limited to biopharmaceutical, is that there are some challenges. Okay, so a great example of biopharmaceutical is the mRNA vaccine that many of us received during the pandemic. And this was an article that was published now about a little over 3 years ago, and we all heard and experienced this very big challenge that we had to vaccinate the entire world, right? And if you read this article, basically the summary is right here. The challenge of making them is unprecedented. So it's the manufacturing challenge that really put a bottleneck in vaccinating the entire world. And in fact, this was a very unique case where the demand was so high that manufacturing couldn't catch up to it, right? But even before this pandemic, so this is an article that was published in the UK in 2017. Okay, here there was a vaccine shortage for hepatitis A and B, and this is straight from FDA website. Even in 2023, we are experiencing shortages of many types of drugs. So this is not necessarily limited to biopharmaceuticals, but all kinds of pharmaceuticals. Okay, so you can imagine that these biopharmaceuticals, we could have instances where shortages become a problem. So let me ask you, what do you think is the biggest reason behind the shortage here of biopharmaceuticals? Doesn't make money, very good. So it's economically driven, actually, financially driven. Okay. So I thought that the reason that there is shortage is because of the raw material, shortage in raw materials. Okay, so that is a big reason. But if you look at the two biggest pies here, this one and that one, those two have something to do with manufacturing, actually. Okay, manufacturing. And financial reason is another big one, actually, especially in case of vaccine, because vaccine is not a big money maker unless you have to vaccinate the entire world as we just seen. Okay. All right. So what's the sort of the problem with biopharmaceutical or pharmaceutical manufacturing in general? Okay. And many of you know this better than I do, but in pharmaceutical industry, products are typically produced using batch processes. So batch processes, if you're a chemical engineer, you learn this, right? Is a process where you put the material into your reactor, right? And then you close it off, and then you run the reaction, and then once the reaction is done, you pull your product out. So this is the batch reactor, batch process. And this is typically how pharmaceutical or biopharmaceutical products are produced. So when people talk about producing pharmaceutical products or drug products, there are really two things that you have to keep in mind. One is that you're creating drug substance. So in case of mRNA vaccine, that might be the mRNA, okay? And then you have drug product where you create something known as formulation. So typically you would have to synthesize your molecule, you purify it somehow, and then you formulate it so that it ends up being some kind of a final product. So the problem with this batch manufacturing is that obviously because you have batch after batch after batch, there could be variations between batches, okay? And oftentimes what you have to do is you're stopping after each step, so you have to test your material. And a lot of times what happens is you have to ship your material into another facility where it's processed using another batch process, okay? So there are a lot of issues. Scale-up is another issue. So let me talk about these problems a little bit more. So in case of a COVID vaccine, okay, there's a very nice article in New York Times. This was an article, I recommend that all of you go take a look at this article, April 28th, 21. And it's got lots of videos, and it's like a perfect article that shows how COVID vaccine was manufactured. Okay? So if you read this, you'll see something that's quite interesting. So in order to make, this is all washed out, but there's a map of USA here, okay? Just imagine that there's a map of USA. So you start with DNA, which is the template for mRNA, okay? Now, what happens is DNA is made in Missouri. And then it's shipped to these two locations, okay? And if you read this article, it's got lots of movies. It's got many movies like this where people are packing stuff so that it gets sent to the next facility, okay? So that's very inefficient, right? You have to test it, make sure, and when it gets there, you have to make sure that the material didn't go bad. And then mRNA is made, for example, in Andover in Massachusetts, okay? And then once mRNA is made from the DNA through in vitro transcription, it's shipped back to, shipped now to Michigan, and some of the material gets shipped back to Missouri so that it gets tested. Okay, and then guess what happens? You pack it again. Okay, once the mRNA is synthesized, it gets now shipped to another site. Okay, to do the formulation. So think about this. This is why it took so long to get the mRNA vaccine out so that we could get vaccinated. Okay, this process takes about 2 months. Okay, there are a lot of testing done in between. So this is a big problem. Now, another problem, the last one that I had on my list was the fact that that there's a scale-up issue, right? So many of us here work at universities, and what we do is we typically work at this scale where we're using round bottom flask to do our discovery. Okay, but once you find something that's good, that has the right activity or functionality, what you want to do is you want to now put it through different tests, right? So in typical pharmaceutics, what happens is you go through these different clinical trials. Okay, and as you go through different clinical trial before you get to the market, what you have to do is you have gradually increased the production rate, volume of production. Okay, so what you do is you go from something called lab scale to something known as pilot scale. So if you're an engineer, you've heard of this word, pilot scale. And then once you succeed at the pilot scale, you go to really commercial scale. Now, one of the reasons that my discipline, chemical engineering, exists is because we know how to do this very well, but this is not always simple. This is called tech transfer. Now, something that used to work in round bottom flask will most likely not work at the pilot scale. You have to figure out the parameters again. And something that used to work at pilot scale, when you go to large scale, again, won't work very well because you have to figure out all the processing parameters. Okay, so this is the scale associated with going from something that you discovered at the lab scale to the market scale. Okay, the scale-up issue when you're relying on batch processes. So FDA has recognized this problem for a long time. Okay, and this is a straight from a fairly high-ranking official. I forget exactly what her title was, but she was like within number 3 of FDA. And she basically says, what we are using in pharmaceutical industry is archaic, and we need to go to continuous manufacturing. Now, it's really only, well, I shouldn't say only because I don't know, but pharmaceutical industry is one of very few industry that heavily rely on batch processes. If you go to chemical or petrochemical industry or even semiconductor industry, most of the manufacturing is done through continuous manufacturing. Okay, and so if we were to transition from the batch manufacturing to continuous manufacturing, how do we do that? So continuous manufacturing is where you have raw material that gets continuously fed in. Whatever process happens, for example, reactions, conversion of the molecule happens continuously, and then you get your product continuously. So there is a continuous flow in and out of your raw material and product, and the system reaches something called steady state. Okay, things don't change as a function of time. So this particular example is called continuous stirred tank reactor. So if you're a chemical engineer, CSTR means something to you. Another way that is very effective in enabling continuous manufacturing, especially when you're dealing with pharmaceuticals, biopharmaceutical, is to use something known as plug flow reactor. Okay, so literally you have pipes. Okay, and we have really world experts in flow chemistry here. But what you have is you have pipes, and the reaction occurs while the material is flowing through the pipe. Okay, so this is how one may use continuous process, flow processes to enable continuous manufacturing in biopharmaceutical or pharmaceutical industry. So one could imagine that instead of having those huge, large volume units where you go from one process to another process and then you have to stop. There's human intervention. There's always, a chance of contamination, something going wrong. What you can do is you can now tie all these different units together so that, raw materials goes into your synthesis reactor and it flows through different units unit operations such that you end up with your final product. Okay? So because you don't have to stop anything, actually this saves you a lot of time and also enhances the reliability of the process. Okay? Also, these processes, because you're continuously doing it, the system itself tends to have much smaller footprint. What does that mean? You can distribute these types of systems in many different places, unlike the example that we saw in mRNA vaccine where they had huge batch process facility scattered in different places, but one facility is centralized, right? That's why you had to ship all this stuff from place to place. So continuous manufacturing is conducive for distributed manufacturing. So those are some of the key advantages, and I believe many of the speakers that will speak after me, and also when we move over to Busan, are people who have really made seminal contributions to all these different processes in flow chemistry. What I do in my lab so far is really just focusing on this right here. Okay, formulation. So somebody has made a drug substance for us, and we're going to take that and then formulate it into a dosage form that can be used by medical professionals. All right. So what are drug formulations? Okay. So these are actual dosage forms that are used by medical professionals so that the drug gets into our body. Okay. So they can be solid formulations, liquid. These are injectables. These are sort of cream-like, something that goes on our skin, okay? And depending on the drug substance and the organ or tissue that you're trying to target, you have to find the right formulation. Now, if you look at these formulations, for example, let's take a look at this injectable. So mRNA vaccine was an injectable, right? And you take a look at what's in there. A lot of times, these formulations have some kind of particles in them. Okay, why? Because we're talking about these fairly sensitive biomolecules in case of biopharmaceuticals. So what you want to do is you want to protect them, number one. Okay, you want to protect them. And a lot of times, where those biological molecules end up is determined by the nature of these particles that encapsulate your drug substance. Okay, so my group has been doing some work on how do we create these particles using flow process in a way that it becomes very reliable. At the same time, how do we accelerate the discovery? And how do we accelerate going from discovery to manufacturing? All right, so let's take a look at mRNA vaccine once again, okay? So we have mRNA, which encodes, right, the protein unit that you want to produce into in the sequence of nucleotides or codons. Okay, this mRNA molecule itself is very fragile, so it has to be protected by something called lipid nanoparticles. And these lipid nanoparticles do two things: it protects the mRNA, but it also determines where these mRNA ends up, to which cell or to which tissue, as you can see here. So clearly, doing formulation in correct way is very important to enhance the efficiency of the drug that you're trying to deliver. All right, so as you saw, there are these micro-scale particles as well as nanoscale particles. So let me show you how people do this right now in industry. So a typical process might involve making a mixture of water and oil, and you agitate it, at high rate, and you end up with emulsions like this. So this would be water droplets in oil. So this is called water and oil emulsion. Also, you can turn these into solid to make solid microparticles, or there are these spray drying process where you literally take a solution and then you spray it so that those little tiny droplets become particles, or you can take big particles, chunk of material, and then you can grind them. Okay, so that you end up with these microparticles. Now, this works okay, but if you look at this, you'll see that immediately the particles are extremely heterogeneous, right? They don't look that pretty. There are some small particles, some large particles, and this sort of works, but if you're thinking about, delivering and having a very precise control over the delivery, this is not necessarily what you want. What you want is something that's very uniform, okay? So these are microparticles. Let's take a look at how mRNA nanoparticles were made. Again, this is straight from that article in New York Times. Okay, and this is a quote from engineer, I guess, at Pfizer. Okay, so take a look at this. They knew that what they were doing to make those lipid nanoparticles was not an ideal solution. They knew that it was inferior. They knew that there was something that was much better, but they just didn't have time. They didn't have time to develop this so that it can be used for manufacturing precise nanoparticles with much higher functionality, okay, and efficiency. So what would that be? Okay, and what are some of the methods that would enable production of homogeneous particles? So I always like to show this to people. So this is a leaky faucet, okay? And if you collect all these droplets, it actually turns out that the volume or the size of these droplets are exactly the same, okay? And the reason for that is because the size of this droplet is determined by balance of two forces, okay? So when the droplet initially starts to grow, it stays on the tip of the faucet because of a force called surface tension, right? And as the droplet gets larger and larger, it gets pulled down because of the gravitational force. So the intricate balance between the surface tension and gravitational force will determine how these droplets are. So that's why they're always the same, okay? So what people have done in about 20 or so years ago is, they tried to basically use similar principle to create these very uniform droplets. So this is a capillary that has been pulled so that you end up with this tapered nozzle that's been inserted into another capillary, and I flow oil and water, okay? And you end up with this situation where you form these very nice droplets one at a time, just like leaky faucet. And here, what you have is you have intricate balance between surface tension and something called viscous force. So there's a liquid that is flowing outside that pulls the liquid, this droplet, off the tip of this nozzle. And it's the balance between these two forces that determine the size of the droplets. So that's why your droplets have such a nice uniformity. Okay? So people have been able to now, take this and make these very nice uniform droplets to convert them into solid particles. I'm going to talk a lot about these things called double emulsions. So they are drop in drop. Okay, so it's water drop dispersed in oil in water. So people call this water in oil in water, double emulsion droplets, drop in drop. Okay, so these microfluidic devices are very good at making very highly uniform emulsion. And these are the precision mixing device that the Pfizer engineer was talking about. Okay, so these are microfluidic mixers. So the dimension of these channels are typically 100 microns or so. And it brings different types of solutions together to create the lipid nanoparticle. And studies have shown time after time that particles made through this method is much more uniform. It has much better encapsulation of mRNA, and they function much better when they're injected into our body. Okay? It's just that people couldn't, figure out how to scale this up in such a short amount of time. All right, so let me talk about these double emulsions a little bit more. Okay, so I said this is drop in drop, so this is water in oil in water, double emulsion. And the reason that I really like them is because I can put stuff inside in these core water droplets. A lot of the biopharmaceuticals are naturally stable in water. Okay, so if you want to deliver, for example, protein, okay, these microcapsules are very, very useful. So you can take double emulsions, you can convert them into solid particles, and that's what this looks like. Okay, so in this case, we added some fluorescent dye inside just to show that we can get really nice encapsulation. And this is really, if you look at it, this is really the very basic structure of the living cell. Living cell is exactly a water compartment, right, encapsulated by a lipid layer that is oily, that's dispersed in water. So these double emulsions are just like the cell. Right? And that's why they're ideal for encapsulating different materials. Now, I was making these, capsules now, that's more than 10 years ago. And there are many reasons that we do this, okay? Once you encapsulate some active ingredients, you can now use different types of external stimuli to induce the release of these materials. Okay, so you can use, for example, heat to break the capsule open so that it gets released. Okay, so that could be very useful. Now, these my collaborators showed up one day and said, Daeon, can you make capsules that will respond to mechanical stimuli? Okay, and the reason you'll understand this very quickly is because they're from orthopedic surgery department. Okay, and what their interest was, can we make a drug delivery particles that can work in this environment, in these joints, where there's a lot of mechanical cue. Okay, and what they wanted to do is, if somebody has, their cartilage, operated on, can we put some particles in there so that as person, progressively puts more and more weight onto it, it breaks capsules open and induces release of some kind of a growth factor that would enable and facilitate healing. Okay, so this is the scenario, right? Can we put something in there? Because usually when people try to put some kind of a growth factor, it gets cleared very quickly. Okay, so you'd have to, keep injecting your knees with these needles. Can we just do it in a way that we inject these particles in there so that they break open? Now, this idea, may not sound so creative because a lot of industry has been doing this. Okay, food industry, clothing industry, for example, have been putting, things inside. And when you chew on it, it releases things. So chewing gum is a great example, right? So that you taste something that was encapsulated. It just turns out that biomedical community has really never taken advantage of using mechanical cues as a stimulus to induce release. So we were able to, quickly show that we can make these very nice capsules. Okay, and then if we put them, put some stress on them, put some load, you can break them open, which means that we can now induce release of that growth factor that you may want. Okay, and we had a lot of fun, putting these into different situations where we can put these capsules onto wound dressing, so that if the wound dressing goes over, your joint area, just moving your joint would induce the release, rupture and release of the active agent. This is an example where we can put the particles into some kind of a patch. Okay, so that, and this patch was for disc herniation actually. So it goes into the disc, and the release of the agent facilitates the healing when the disc is injured. Okay, so that's all good. So we know how to do this very well. But let's go back to that issue that I was talking about: manufacturing issues, scale up, and so forth. How much time do I have, by the way? I don't want to go over the time. 15 minutes. Okay, perfect. So here's something that people don't talk about when people talk about microfluidics. So this is a graph that my student generated for me. I used to tell my student, hey, I need large volume of my sample for my collaborators. So go into the lab, start your microfluidics, go home, come back next day, and then you'll have your material ready. Okay? That's what professors do, right? And then he says, no, that doesn't work that way. I have to be there. I say, what's the problem? Why do you have to be there? Because he says, something happens if I'm not there. So this is the data that he got for me. So he kept the flow rate constant. And he looked at the droplet that was being generated, okay, over 8-hour period. And this is what he was talking about. There are, times where things work quite well so that we can publish papers. But if you try to do this for hours and hours, things happen, okay? This is double emulsion formation where even though it's exactly the same flow rate, sometimes you get very nice double emulsions, sometimes you don't. And we don't know why. We don't know why, okay? So that's why human has to be there, okay? And an experienced human has to be there. Now, in a lot of cases also, what we realize is that we want different types of capsules. Sometimes we want small diameter, large thicknesses. Sometimes we want more reagent inside. So we have to create these one by one, fighting all these problems. So that's a problem, right? What we want to do is we want to create a library of particles like this so we can do screening and discovery quickly as possible. But preparing a sample like this would take weeks, if not months, for an average graduate student. Okay? All right, so again, this is an example where we have this is the ideal case, but a lot of times we end up seeing things like this. Okay, and I won't go into the details, but you can see that these are all very nice, but you see like things that are happening that doesn't look good, right? This is a case where even the inner phase has escaped out of the middle phase, so you don't even form nice double emulsion droplet. Okay, so how do we deal with these problems? This is where the recent developments in machine learning and AI really helped us. Okay, so first of all, we have to teach computer, if its computer is going to solve this problem, how to recognize these problems. So we looked at how autonomous vehicles drive themselves around, right? And there's this convolutional neural network that detects different objects when it looks and takes images. And then it can assign, those are cars, that's a person on bicycle, and so forth. So we use the exact same technology. It's just that we train it to look at our droplet generation formation. Okay? So like you give an image like this, and it will say, oh, there's a jet here, there are nice droplets, and this droplet looks larger. So you're not in the right regime. Okay? So we are using something that has been used in completely different field, but we are now transferring that to our microfluidic system. So the way we do this is we take lots and lots of images, and we train our convolutional neural network. So that it learns, okay, that's right, all those things are not right. Okay? And once it recognizes that something's not happening in the correct way, it adjusts the flow rates and so forth so that it brings it back to a system where it's generating nice double emulsions like this. Okay? So like for example, take a look at this. So nice double emulsion droplets, and then something happens. The student didn't do anything. Okay? But the instrument immediately tells something happened, and it starts to change the flow rates and so forth, and I won't go into the details, so that it brings it back to the nice double emulsion generation regime. Okay? That's what autonomous cars do. When there's a problem, it immediately reacts, right? And the reaction time is approximately 150 milliseconds. This is faster than a very experienced F1 driver responding to some kind of a sudden event. Okay? So we now put together this whole thing where we have our double emulsion generator, and we have a neural network that can sense when things are going wrong, and we now have feedback control loop that will basically tell the instrument to change the flow rates, and while it's doing it, when it's creating materials that doesn't look right, it basically selectively directs the product either to the waste or your collection vessels. Okay? So this is a system that we put together in the lab, and the goal is, see if we can prepare a library like this, where we have different shell thicknesses, and then we have different concentration of some kind of a biopharmaceutical that gets encapsulated. Okay, so I think, let's see, this is a movie that my students put together to show you what it looks like. So music is very important here. Okay, so this is the graphic user interface. You will tell the instrument what you want, okay? You will tell, okay, I want these thicknesses, that's my particle size, I want my drug to go from this concentration to that concentration, and I don't know if the music would be playing in the lab, but just assume that it is, okay? And then you connect the camera. All right, and we are gonna start the process. All right, so this is your device, and your device doesn't have to look exactly the same every time. The computer algorithm is smart enough to know that you have a device, and it will do this. It will tune things, it will tune the flow rates and so forth, right? And then once it looks right, it will start collecting. It will start collecting, and we have a collector, okay, that rotates so that only the right droplets gets dispensed into the vessel. So the 25 types of double emulsions that I told you, it would take normally weeks to make, can be made in a few hours using this technique, and you don't have to be there. Okay, this is all done by the computer and the instruments. All right, so very good, very good. So these are the droplets that we can make. They're beautiful. So that should hopefully accelerate the discovery process. Okay, and human intervention is minimal in that process. Now, the thing with microfluidics is that it's great for discovery, okay, especially if you use what I just showed you. But the problem is the production rate is extremely, extremely low, okay? This is if you're doing well, okay? So if you want to create, say, 100 liters from this flow rate, it will only take 11 years. So that's why those Pfizer engineers didn't even think about using the precision mixing device, okay? Because it would take 11 years, and a lot of people would be dead by that time, right? So the thing with this particular type of flow chemistry that uses microfluidics is that there's very low production rate despite the uniformity that it gives you. Okay, so there's this gap between what's being done at the discovery scale at a lot of universities and what the industry requires. Okay, so one simple idea is, okay, what if I just operate many and many of these? Okay, that's what I would do, right? The problem is then you need many and many fluid handling units and many tubings and pipes and so forth. That's not great. That's a very inefficient way of operating a manufacturing facility. And that's how old computer used to work. You had many operators that were just pulling and doing all kinds of things manually. And unfortunately, that's what a lot of companies are doing that right now. So we sort of didn't tell you which companies these are, but they basically put many microfluidic devices and then put many pumps next to each other. Okay? But let me show you how we solve this problem. So we are, and I work on this with my good colleague Dave Isidore, we are inspired by microelectronics industry. So let's say this is a single microfluidic device that makes beautiful droplets, but it's done at very low flow rate, right? So microelectronics industry, in 1940s made this first transistor, which was about this big. But we now have these integrated chips in our computers, right? That have millions of transistors on a single chip. How did they do it? They miniaturized it, and then they did something called very large-scale integration. They parallelized it. They put a lot of these mini transistors onto the first floor of this multi-layer structure. And instead of needing million inputs and outputs, they have small number of input and outputs, and the electrons are shuttled in and out through these things called lines. Okay? And these lines are connected to these transistors through something called vias. So here they figured out how to distribute electrons among these many different transistors. So we do the same thing. We have many droplet generators that gets parallelized, just like the same principle that's used in VLSI, okay, by bringing in water and oil through a few channels and distributing it between different droplet generators. Okay? Now, when you see this, you should be very suspicious and because I'm telling you that flow rate here will be exactly same as here and there, and that doesn't make sense. Right? That doesn't make sense. And that's where you had to do a lot of engineering. I'll talk about this in Busan more in detail. But the secret behind this is just like if you had a shower room like this, okay? And if you want all these shower heads to give you the exact same flow rate, the secret is behind the wall. Okay? You want the piping behind the wall to have diameter that's as large as possible, so the pressure drop there is very small. Okay, compared to the pressure drop of each shower head. So it's the exact same fluid mechanics equation that you solve, but now at the micrometer scale. So what we can do is again, borrowing the industry inspiration from microelectronics industry, we create, this is a 4-inch wafer, and we now have 20,000 droplet generators, okay, that are parallelized. We have one input for water, one input for oil, and these are for products. Okay, so we just need 2 pumps or 3 pumps if it's double emulsion. Okay, and this is what it looks like. All these 20,000 droplet generators are all making the exact same droplets. Okay, and if you actually play this, now we are generating these emulsions and particles at the scale that is relevant for pharmaceutical industry. It's perfect for pharmaceutical industry. Okay, so this is tens of liters per hour. And remember, this is a continuous flow process, so you don't have to stop this. All right, so we know how to do this with double emulsions as well. And we know how to do this with lipid nanoparticles, which are the particles that would encapsulate mRNA, so that we can use the same device to do the discovery. So this is one single device to do the discovery. And once you figure out the device geometry as well as the solution conditions and processing conditions, all you have to do is copy and paste this at different scale, okay? So that the fundamental unit that makes your particle is not changing. So nothing changes as we go from very small discovery scale to manufacturing scale, but you're changing the scale at which you're making your material. Okay? So I'm going to conclude. Hopefully I didn't go over my time too much. What we can do is we can now use, AI and machine learning to have a better handle on creating materials from these microfluidic devices. I showed you one example, but you can think about many different examples. And then you can do your own testing. And because you have high quality and you have library of many, many different types of samples with high quality, you'll now get large data set that's high quality. And then you can again use AI and machine learning to go through this loop very quickly, hopefully, to accelerate your discovery. Now let's say you found that this particle works very well with this device. What do? You need to go through these different clinical trials, right? All you need to do is you need to just parallelize depending on what scale you need. Individual droplet generator does not change. Your particle will not change. So this is easy. Okay, this is easy. So I was very proud of this concept, and then I saw this video. Everybody know who this is? Yeah, who is this? Steve Jobs. So he gave a lecture at MIT many, many years ago, and listen to him.
Well, we don't hear permission, but yes, I can. Here's how it works. One of the things we do is we actually, when we want to build an engineering prototype, what happens in one of the key things that manufacturing can contribute to competitive advantage is time to market. Why is that? Because the way most things work is you design your product here, and after you're done, you throw it over the wall and you design your manufacturing process here, right? Sorting out a bunch of things that maybe weren't done right here, fixing them, changing them, and then completing the process design. What you want to do is do this and ship it right here while your competitors explode. And that's what we've been able to do in many cases. What we do is we suck data out of our CAD system.
So apparently, Steve Jobs knew this long time ago, and that's how Apple was able to flourish. Okay, they were developing their new computers and processes on the means of manufacturing. So nothing changes when they go from discovery to manufacturing, and I believe we can do this in the pharmaceutical industry as well. With that, I'd like to thank everyone for your attention. Thank you.
Okay, thank you for the really time, like some peeking to the sun schedule. So, but even though we have some panel discussion 40 minutes, but you can make a question now if you have. Please.
Very beautiful talk.
Thank you.
And, I'm the quite beginner in this area, so I do not know about the detail. You really nicely modified conditions and could really obtain nicely regulated size control emulsion and by using the AI. And what I thought is why that really changed on the uncontrolled flow is generated. Because even if they're using the very pure substrate and very pure solution, the very controlled flow rate, even that you could not really control the size, control size of the emulsion, and so you had to really modify.
Yeah, the question is why do I see this? Okay, so the answer is we don't know, and AI doesn't care. But as a scientist, I want to know, right? I want to know. So things like small vibration matters, okay? If somebody walks by, okay? Because these are very sensitive. These are, flow at very small scale, okay? If there's an earthquake in Japan, right? Something like this can happen. But just somebody walking by, sometimes a bubble may get trapped in places that we don't see, okay? And that's going to cause an issue because bubble is compressible, okay? So what we had to do actually, and this is really fun because the way we fix this, and I showed you how now AI can fix this problem, right? Let's see. My computer is stuck. Okay, here we go. So you know, it entered into the wrong regime right there. The student didn't do anything, but it recovers very quickly, right? So when you're watching TV, and if TV doesn't work very well, what do? You turn it off, turn it on, or you tap it, right? That's exactly what we did. This is tap. Okay, this is flow rate pulse. And then see if it corrects itself. Pulse again. So it kicks it until it starts working again. Now, we don't know why it got into the wrong regime. We don't know. That's a great question.
And also, like, how did you really improve that situation? Just you control the flow rate?
Yeah. Kick it. Yeah, usually, you know, changing flow rate in different ways will fix problems.
So that you already know that, or if you really see the jet flow, you can really reduce or increase.
Exactly. But in this case, right, this is purely inspired by what experimentalists do in the lab. A lot of times what we do is we tap the tubing, we tap the stage of the microscope, okay? And a lot of times that does it. We don't know why. It's probably because, you know, when you look at these different types of regimes, you know, if I'm looking at this as like a free energy landscape, I think there are multiple minima. So the system gets trapped in different minima, okay? And then there's one minimum that's good for you, and you want your system to go there. But it often gets trapped in minimum that's not good for you. So you have to bring it out of that. So you have to put some energy in to bring it out.
Thank you so much.
Sure. Very, very good question.
So any? Okay. Professor Lee, thank you so much for your
Sure, thank
talk.
you.
I'm really interested. I actually searched your paper, Chemical Engineering Journal, about this kind of CNN paper. So one of my kind of curiosity is that size of the droplet that you make, really important factor that you want to check. But in case of, for example, we consider about the encapsulation of some drug or biopharmaceutical. So size is important, and also encapsulation of some biopharmaceutical material inside also is important. So your algorithm try to fix the size by tapping or changing the flow rate or this kind of thing. But how we are sure that the other objective, like encapsulation of the drug or other products.
Very good, very good. So in our case, the question is, how do we ensure that the concentration of drug is what you targeted? In this case, it was easy because our drug had color. So it was not a drug, it was a dye, right? So you can read the grayscale value. But if there's an inline detector that can sense your molecule, like RNA, in case of RNA, it's there's a UV peak that you read, for example, at 260 nanometers, I think, okay? You can basically do the detection. The way we control the concentration is we have two different pumps that deliver pure water and a solution of known concentration, and then we mix them in different ratios. So that's how we ensure the concentration.
So as a result, if there are multiple objectives that we want to control together, either the system will be more complex, but anyhow we can somehow.
Absolutely.
Thank you so much.
And it does that like in 150 milliseconds.
That's fine. Thank you so much.
So you make it some 20,000 droplet generator, and then there's some of the fabrication technology transfer to the pharmaceutical industry.
I have a company that is trying to do that. Yes.
Now it's on the negotiating.
Yes, we have been talking to a lot of pharmaceutical companies.
I see. Okay, go ahead.
Yeah, how to monitoring and sensory and control every microchannel unit when you scale?
Very good. Okay, so you asked a question that I ask my students all the time. So I know how to do this feedback control with one channel. I don't know how to do it with multiple channels yet. I don't know how to do that yet. But that's where we need to go, because that enhances quality assurance during manufacturing. So that's extremely important. We have some ideas, but that's what we'll work on.
Yeah, I think
Wonderful question.
So many camera in there.
Yeah, I don't think that's the way to do it. I don't think that's the way to do it. I think there are different ways of doing it.
How fast to respond?
150 millisecond.
100 millisecond.
Now, okay, so the instrument knows that something's wrong, in 150 milliseconds, but then it has to respond, right? We may know something's wrong, but our body has to respond. So same thing with our instrument. And that's usually a few seconds. That's seconds. But the instrument knows something's wrong in 150 milliseconds. So every 150 milliseconds it's making a decision.
Yeah. What kinds of the main cause the projects, the different kinds of drug shape? In my sense, maybe pump. Yeah. I think all kinds of pump cannot give us the correct.
That's right.
Yeah.
That's right. Yeah. So syringe pumps that people use typically in the lab are not perfect. So what we've learned is that if you do pressure driven, it gives you much more stable flows.
Yeah.
So we do that as well. We do pressure driven flow.
Okay, so I think the same topic is he's going to give a talk in the Busan, and so he's going to be
It'll be more technical too.
release the more secret, right? So we have some chance to learn about it. Okay, thanks to the speaker team. Okay, second speaker is from Australia, and that is my long-time friend, Volker Hesses, right? Actually, he's working on some German, and then now he's working on some Australia, University of Ireland. Did you enjoy some of the travel with your wife in Japan?
I enjoyed it a lot. It was wonderful. It's wonderful to be here. Thank you for the invitation. Yes, we'll see if we get started. We have to go to the first slide.
Capillary forces are stronger in flow chemistry channels than gravity forces. So every experiment you make is a space experiment, and many experts say that flow chemistry is a future for making chemistry operations in space. And we need a lot of chemical operations in space if we believe in what NASA and all people say, that we will have villages on Moon soon, and that we will have a village on Mars within the next 20 years, and we need chemistry. So I was born with microreactors. Yeah, they were invented in 94 or 92, whatever you say, yeah, FZK and PNNL. So I went to the Institute of Microtechnology, and we were the first to make the technology commercial. Yeah, it was SK Corporation here in Korea, it was LG Chem, it was Kyoto in Japan. They bought our microreactors, and people said, Professor Rio, not to forget, sorry if I forget somebody, and people said they are small, yeah, and said no. 300 liters power in this device. We brought it to Angewandte Chemie. Now, after some time, flow chemistry is 20 years old. Microfluidics and microreactors are 30 or 35 years old. So I looked for new frontiers. Luckily, when I came to Australia, they built the space agency just 200 meters next to my office. So I thought I should make space research. I'm professor in England. This is Leicester. This is a national space center over here. I led the first Australian space experiment, NASA ISS experiment, and this is an official photo. This is not a fake. This is a real photo. It's microgravity, so our sample is flowing. And this is a very recent paper about sustainability flow chemistry. So I still do flow chemistry, you know, not to forget, but space is an interesting topic. So I have to see, I don't see anything. Ah, yeah, good. Okay, going to the next slide. I also made some, this is a talk to the public, so I made some spacey, catchy pictures. Now, okay, so 2030 is the Moon, 2040 is the Mars. We have got a nice project in Australia, and I have to advertise for this, from ARC, Australian Research Council, which is Plants for Space. This is a center of excellence, and this goes 10 years, 2033. I even thought when I got it, will I still live there? Because I turned 60 this year. You cannot necessarily assume you got 70. So it's interesting. I never had a project so long. I had European projects 5-6 years, but 10 years project, that is good. The project has $35 million. We already earned $15 million in 1 year. Yeah. So when you have such a project, it's like a stamp to print money. It's really a very good tool of the Australian Research Council, and we work with NASA and many others, as you will see in a second. And I'm a program lead for the engineering to make products, to make the manufacturing biomanufacturing in space, and I will talk about this. Now, when you think about space, we have an artist, which we pay. We pay him full time because it's important that people know how space looks like. I'm the research director of the Andy Thomas Center for Space Resources. If you don't know who Andy Thomas is, he's from Adelaide, by the way. He's the Neil Armstrong of Australia, so the first astronaut we had in '73. He's still living in the US. So you think about something like this. It looks gray, dirty. It doesn't look like a nice place to be. A lot of robots, artificial intelligence, cybersecurity, habitats like in Antarctica. This is how you typically assume space. But we want to make long-term human presence in deep space, and you have to have fun. Like I have fun on this mission here with COFS. Yeah, I have a lot of fun. So the question is, what is missing? And we analyzed this in a very recent paper. We looked what all the NASA missions and Elon Musk missions, here SpaceX mission, that are recent missions. What did they do? And the color coding is here. Yeah, so don't look on this color. Look on that color. And you see they did a lot for technology, but they didn't do much for humans. Yeah, my wife, when she came to Tokyo, she knows what to buy. Channel, Zara, she knows where to go. When you fly next time to the moon for weekend shopping, do you know now what to buy on the moon? So we miss space brands. We miss products made by microreactors in space. And we gave answers to this in this topic. What is the tariff on moon, the currency? Is it US dollar? Is it won? Is it yen? We gave answers to this. You have to answer this. You cannot make a space presence and just look for splitting water to hydrogen. That is not enough. And you have to give other answers for investor rights, no-take zones. Everything which is green is from our work. So that is where we want to fill gaps. We admit NASA, JAXA, they do all much better in the classical fields as Australia can do. Australia has to go in the soft parts of human well-being. Australia has to lead the chemical manufacturing. Yeah? Here, for you, very obvious here. What is done over here, that's a picture of me 10 years ago. That's my student, Don, who made Australia's first space experiment. And she made the second space experiment, which I all will show, and this is the Mars outside. So that is our vision. Our chemical factory should look on moon. Now I'm doing plant growth. I never could imagine I come from microreactors to plant growth. That is the vision for vertical farming in space from our ASC center, which I mentioned. So what do we do there? We do plants, clear. Plants make foods and flavor. That's still very clear, and it's no further explanation. But we reprogram by breeding and by DNA modification plants, so they are biomanufacturers. Plants which make only caffeine, nothing else. Yeah? Caffeine is a medicine and other medicines. So that is what we are doing. We reprogram plants, they are doing biopolymers to make houses out of it, like you make from bamboo houses. And we look for recycling. That is my engineering job over there. We have to look for space law. Who owns the moon? Who decides what is a legacy? We saw FDA. Who is the FDA on moon? That is all not decided. And we have to work very strongly in teams and probably in international teams. And this is a slide, and I'm happy Gioia gave it to me. She's a lead investigator from NASA for plants. Why do we grow plants in space? First of all, food. That stands to reason. But second, that is my argument, it makes us feeling well if we see plants growing, not only the eating, and then she used a NASA term, this is enclosed environmental living chambers. Yeah? So engineering systems, you could talk. So that are NASA's advanced space habitat. Yeah? So that is what we want to develop. And I will show you in the second part what flow chemistry does over here, if you miss flow chemistry. Now, what are our missions? We want to make plants which are zero waste. Plants which have maybe no roots. Yeah? If we think roots are waste, we omit roots and find another way the plant take up the nutrients. Or we find a way to use the roots. So that is one of our missions. And you have to see there's $35 million behind, and then there's maybe 3, 4, 5 groups working on this. So the next mission is, where is it? complete nutrition plants, plants who can give you all nutrients which you need. You don't need meat anymore. On-demand bioresource production, I talked this, making medicines, making bioplastics. And we also want, I'm old, not too much worth to invest in me anymore, but we want to make ready the next future workforce of Australia and society and also globally. And we have here 4 or 5 people, full-time equivalent, who work only on this. Yeah? So this is not just a saying, there are 4 or 5 people behind who have the mission to make that happen. Now, I come to the enclosed systems. We have them on Earth. Yeah? We don't grow plants only on acres. Yeah? We don't only grow them on fields. If you go to Netherlands, Netherlands is the most productive agricultural country in Europe. Yeah? They sell all foods to whole of Europe. Netherlands are so small. So how can then they do it? They have process intensification in terms of vertical production and production in glass houses. You see there is a level of engineering complexity which goes up here that has advantages and disadvantages. I don't want to discuss them all. In US, around NASA, in Florida, and all the southern states, there are clever people who think about a kind of taxonomy of those systems. I just want to mention this Bridges Consortium. Yeah? You can read what it basically means. And this is like automation and modularization, future factories which we have for the flow chemistry. Now, what is the vision again? Thanks to Joya, you can have your enclosed chamber outside the living area where the plants are grown. You can integrate it here, like when you go in a cafe in Singapore or whatsoever, where you have a whole biophilic design which makes you happy when you enjoy your coffee or your pizza, whatever you do over there. So biophilic space design is one PhD which I have only on biophilic design. Now, there is a vision of NASA. That's the last slide I think I show from Joya. There's a vision from the ISS, which is now our main laboratory, over the so-called lunar gateway to the moon, long-term, short-term, and then there is a Mars gateway or Mars transit to Mars. And there's a vision of the complexity of those greenhouses. This is all given here. I don't want to go into details. I also want to keep my time if I can. So, but just note there are plans, and the plans are more clear here naturally than they are clear over here. Now, that is work from me. That is work in Plant for Space. Our partner, La Trobe University, if you've not heard, it's a university in northern Melbourne. They have a very big greenhouse, and they have a huge pump system. That is what he has shown. There's a distribution by pressure drop to all the cabinets. Yeah, this is one cabinet full of tomatoes, but there are many cabinets, like the shower, which was shown, and that is all fed by one central system. So this is very optimized. Now, I made an LCA on this, and look here at the scale. We start at 99.96%. So everything is electricity here, and that is great. Because what is bad is to use too much fertilizers. Fertilizers is payload, and Gioia from NASA always says, don't bring fertilizers to moon. You have to make the fertilizers on moon, and we do it by flow chemistry, as I will show. So it's good if everything is electricity. Please note this is %. That doesn't mean it's high. It just means the fertilizer is largely eliminated by a good practice. And the moon systems we have currently are the opposite. I don't show that, but believe me, are the opposite. They are fertilizer dominated, and that will not work. So we have to come to such performance which we have on Earth. Yes. Joya made a paper on their NASA veggie system. You know, something we would not care about Earth is in plants, most of the water transpires. You lose 99% of the water by transpiration, the ISS, and it goes outside in the vacuum. Now, on Earth, you can not take care for it. On space, maybe not. So we made 5 scenarios. We said we do not consider it as waste, like the transpiration. We just ignore it, as we do sometimes on Earth. We recycle or we not recycle. Yeah? This is given here, 5 scenarios. And here I look now on what is called the circular matrix, which I also introduced to chemistry and flow chemistry. I think no other group has done that before. It's a great matrix. It's from the Ellen MacArthur Foundation, EMF. And the only thing you have to take care here is the MCI, the material circularity indicator. Very easy. 1 is circular, 0 is not circular. So we analyzed now those scenarios. Now, before I come to this here, I show the results. In scenario 1, where we just are ignorant, we have a high MCI, but because we are ignorant. In scenario 2 is where we are not ignorant, we have a low MCI. And most Earth systems are around 40, 50, 60% circular. Yeah? Show me one which is 90% circular. I would be happy to see that. And in the scenario 5, which is a hypothetical one, we have 96. So where we recycle everything, we still lose something, but it's good. Now this material circularity indicator, what I love about it, it's about product. It's not about waste only. It's a mathematical function, which I don't show here. It has lifetime and product quality without defining it. And if we talk about product quality in food, it's psychology, yeah? The taste, the palatability, how it's called. So we have a psychologue in our case. And I will introduce a new definition for the material circularity indicator based on food psychology. Okay, this is the MCI. Oh. You see, we get attention. This is the number 1 of NASA, yeah? The head of NASA, Senator Bill Nelson, and the number 2, Pamela Melroy, that's a lady here. Where is she here? Yeah, here. She was also there. So we had a high attention. This is the head of the Australian Space Agency, and here we have our regulate rover yards, by the way. This is our space laboratory, you see it. So we are in close contact with NASA and their partner. Now, goal was to make perfect nutrient modeling. And I always enjoy when I'm in Asia, I lose a lot of weight because the food is good here and it's much based on vegetables. So the question was, can a computer model predict which of the vegetables are best for an astronaut? An astronaut has different calories than somebody on Earth. Now we made a computer model and we considered nutrients. You should have all nutrients. You should not eat too much. Who wants to eat 3 kilograms vegetables per day? We said 1.2, and we have to make it caloric because you still want to fit in your spacesuit. Yeah? You don't want to have problems to go in the future. So these were our considerations. And then we made by hand calculations the sustainability. That is all about sustainability and compliance effect. Okay. And finally, we made something which was called a space salad, and I will show you this in a second. Just keep that in mind. So that is the outcome here. Now, we made different scenarios, and we have different crops. And by the way, it's very difficult to get data on crops. They are not perfectly inventorized. I'm really surprised. Yeah? So it's sometimes not known how much B12 is in garlic, vitamin B12, how much vitamin D is known. And the computer is stupid. If the computer doesn't have the data, it doesn't work. So it took us quite some time to get all the data for all the vitamins, which I don't show here, of the crops. Now, finally, when we had it, we could say, this is a great scenario. It gives the astronaut everything, but you have to eat 2.2 kilograms per day. This is too much. Nobody wants to eat so much. So we looked for scenario number 4, 1.2 kilograms. So this is our best scenario. This is a space salad. This is something with meat, with salmon, which makes the issues for vegetables better. But remember this scenario. By the way, you should eat only sweet potatoes. They give almost everything what you need. The rest is just addition to it. So this is something we discovered. The only disadvantage of sweet potatoes is they have to be cooked. The rest could be eaten without cooking. So now when we look at the plant area, we think about sustainability. In a spaceship, we don't have a lot of place. So we have to take care to take plants which grow very fast, respectively take low space. Now remember, sweet potato was our winner. It doesn't take a lot of space, actually. What takes a lot of space is poppy, for example, in this scenario, and peanut. So the things which were not important. So what we learned is the plants which we really need, they have low space, and the plants which we don't need have larger space. So there is something we have to consider. We have to design future plants. who makes that all better. And on Earth, we have a lot of space. In space, there is not much room. The same goes for fertilizer usage. I don't go in detail, but there are big differences between fertilizer usages between the scenarios. Now, it's about food. Food is psychology. You look at it, you taste it, and we made a small psychology study here in New York with somebody who worked for NASA's Venus exploration area, and the outcome was, some said it's good, some it's bad, that is normal, so we learned something about our space a lot. The American Chemical Society, not the journal where we published, the Chemical Society highlighted in an article, and I have no stake in this, they did it on their own, designing the perfect meal for astronauts. I got 100 press releases, really 100, 95 or so, and here you see some of them. I never had such a response from social media. So obviously food, space is highly interesting. Now I come to flow chemistry. Hideki Miyamoto, whom I just visited at the University of Tokyo, gave me an asteroid. The asteroid is here, Campo del Celio. I brought this to Australia and I had to declare it. Yeah? In Australia, they always say, just declare it, yeah? Otherwise you go to prison. So I said, I have soil. And the lady asked me, what soil do you have? I said, soil from space. She asked me, how old is it? I said, 2 billion years. She said, go away and let me through. I was expecting she takes it away and my experiment is gone. Okay, so I brought it to Australia. I took Aquaregia, you know that. And we could make a cobalt extraction with this asteroid, which is published in this journal. So leaching of space materials is a good point. We want to leach phosphorus. It's a macronutrient for plants. We want to leach potassium, second macronutrient. The third one is nitrogen. That's more difficult. So let me talk about phosphorus and potassium. We designed here a circular process. So we leach it, phosphorus, from moon crusts. That's where we start. Then we give it to lettuce, which is a fast-growing plant, and we use the roots, remember, no waste, and we burn it in an oven. We make a leaching of ashes, all in a flow reactor, yeah? So this is a known process on Earth, and we get it back. So it's not a full circular process, but at least a way to it. And that compares well to, here, this is from the West Coast in the US, from researchers who work for NASA, who made a paper here about the circular Mars biofactory. Yeah? So that is similar approach. Now, you can use regolith to grow plants. Plants are much tougher than humans, I always say. You can irritate the plants, bombard them with alpha rays, which we do here, 1,000, 10,000 times what you get in space, and the plants still grow. I'm not sure if I would be bombarded with 1,000 times, if I still could drink a beer in the evening, would be a question to solve. But the plants still grow, and they grow in regolith, in moons. This is moon regolith, simulated, but still quite similar. Here are the biomass data. In the bigger picture, and this is what the great colleagues in the US do in this CHARGE consortium, they convert regolith to soil. They put worms, normal worms, in the regolith, and the worms and microbiome create a normal soil after just a few weeks, so 4 generations. And you see how much the plants grow better when the regolith is transferred to the soil. And we love hydroponics. We say great things about it, but the regolith uses less water, has less power, and produces finally good biomass. So there is a competition between regolith and hydroponics. Now I come to social things. The older I get, the more I'm interested in social points, also for flow chemistry. These are the most remote islands of the Earth. This is Tristan da Cunha, 3,500 here, 4,000 kilometers here. Napoleon, for good reasons, was brought here. So St. Helena is over there. Pitcairn, that is a bounty, if you remember. Those people live since 300-400 years peacefully together, and they don't even have a mobile phone. There's no Wi-Fi over here. They don't know that. And they survive. So we should not look on Antarctica when we look for space, but we should look at those islands. They found ways to make supply chains. To Tristan da Cunha, 1 time per year, a ship comes. 1 time. If you forget something to order, you have to wait 1 year until you get it. This is how we envision it for space. I talked about products. They make in Pitcairn Islands a unique honey. They have no diseases there for bees. And they sell it maybe for 10 times the price. So that is possible because it's high quality honey. I said, what would you buy when you go for weekend shopping on the moon? So we have to have an idea about moon products. And finally, decision making. On those islands, it's like here. We have to see every day into the face. Yeah? If you decide something which does hurt me, we have to look every time. That is different to big nations. And so the decision making cannot be so hierarchical. So we can learn from those islands. So the whole article is about how can we learn from those islands. And the great thing is the paper was put in a machine, in a computer, Excel Pro, digital twin software. And I sent it to the director, and after 10 minutes, believe it really to me, I got this all from the computer. It was the first time I really believe in artificial intelligence. So the computer gave that name. The computer composed a team for decision making in space. And I thought all these names here make sense. The computer even gave that text, yeah, it's from the computer. It's not taken from the web. It's longer, so I cut it. The computer gave a job definition for each of those agents here, for the policy and compliance agent. And finally, what I love as an engineer, the computer gave a mathematical function how to optimize this all with social engineering, conflict resolution rate. And that is called MAX. That is a big thing if you go to Amazon or so, multi-agent generative systems, intelligent digital twins. In this sense, I'm responsible for digital twins in plants for space. I thought my strength is on the experimental side. So my strength is to make physical twins, which are part of digital twins. So I thought, why making digital twins? Let's make a digital triple. 2 physical twins and 1 digital version. So I have a physical twin center, which is a mountain. This is a monastery, 1,000 meters high in Spain, near Barcelona. This is space for me. Yeah? So that is a ISS. And I cannot reproduce experiments on ISS, which is a big disadvantage. So I sent the data via an IoT hub to the Earth in a controlled environment which can reproduce the space data. We have microgravity machines on Earth. We have cosmic ray on Earth. We can quite well reproduce space. So, and that were experiments which we were made under different conditions. Now, question is always, how can you replicate that? And here you have an idea. Yeah? This is 1 day, I think. Yeah? It's 1 day, you see the hours. And when the sun comes up in Spain, then the temperatures rise, and you see we could replicate it. With our symbol, we have really not a lot of money, means quite well. We developed a machine learning model. This is still not great at all, but it's partly valid in some cases. It's not valid in other cases, and we know why. So we work hard on that. We made a second study, and again, this should show you what digital twins could do. This goes to climate 2050. Everything gets harder. So we let lettuce grow first in Lleida, which is near to Barcelona. Then lettuce grows in the next week in Adelaide. It goes to Paris. It goes to St. Louis and to Singapore. And who would actually let lettuce grow in Fairbanks? With digital twins, you can do it. We did it in May. So in May, Fairbanks is also reasonably warm. Now, these are the temperatures. Yeah? Oh. These are the temperatures. This is Singapore, very constant as you expected. This is Fairbanks. And the other 4 cities were almost the same. Yeah? It is strange. I was in Tokyo, it was 23 degrees. Even the locals said to me, unusually warm. So we expected large differences here, but global warming has another opinion, so this is the reality. And we replicated this in the growth chamber. It's not perfect, but again, within our means we could do. By the way, it rained only 3 times in 6 weeks in the cities. There's also something about the change in the climate. This is Singapore, where it's expected to rain. So no rain in those cities virtually. Now, even under those conditions, what came out is, at the end, in Singapore, we have a large increase in biomass productivity. You see it? We expect that, because Singapore is good. Would you have expected a large increase in Fairbanks? Would you grow lettuce in Alaska? That is the out-of-the-box thinking which can come out of using artificial intelligence, these digital twin models. Nobody would go to Alaska to grow, but obviously, at least in May, there was something good for it. Now, in the last part, I come to what was shown before, the Pfizer nano droplets. We use such droplets for making beverages for astronauts. How does it work? We have spontaneous emulsification. We put a surfactant in oil, and it doesn't like oil at all. So once it has a chance to see water, it immediately jumps off, and since that goes so fast, it disrupts the interface, and then we get nanoemulsions. Question is, can the forced emulsification, this is spontaneous, in flow make better results? That is a question, and can we use it in space? Now, here we see, yes, it can. It depends on the surfactant to oil ratio. We don't want this to be too high because when you drink your drink, you don't want to drink only surfactant, even if it is biocompatible, and it's also very viscous. So there's a compromise, and the compromise is here. 100 nanometer, this is enough. We go for cloudy emulsions. That is what we want. Now, when I stand in the morning in the queue at the coffee machine, I'm glad if it's 30 seconds, and I don't have to wait 10 minutes for my coffee. Would you wait 35 minutes for your space drink? You would not. So that is a batch technology. In a flow chemistry device and in the Corning device, you go to 5 minutes. It should be finally 1 minute or 40 seconds, but I'm not a commercial producer. I'm happy enough to have brought it down to 5 minutes, and there's room for optimization. Now, the vision is, and we do this with the Dutch company Battery Automation, to have the personal drink. Astronauts are monitored every day, so the data are there. You even can see it in the web. You can use an interface, and you can tell the machine which drink you have. That is what PepsiCo does and Coca-Cola. They build machines where you can build your personal Coca-Cola and so on. That is possible with espresso today. We will see more of such machines in future. Now, question is, knowing what Pfizer has done, can we use the same nanoemulsions for medicines? You should know people lose 20% of their bone mass in space. That is a serious problem. So how do they work against it? They have a treadmill in space where people are sportive, and they eat a lot of healthy food. We say that's all fine, but supplemented by the nanoemulsions. And that is possible. You see, you have through the helmet, you can inject. There is a lot of medicines are given in space by auto-injections. They don't take only tablets. Yeah, when you walk on moon, you obviously cannot lift off your helmet and take the pill if it's an emergency case, but you can inject it. So this is how it looks like. Now, what my student makes is a mapping here. Yeah? This is oil, water, and the surfactant. And the nanoemulsions obviously forms not only at every composition. So we are happy with this. We need something stable which doesn't jump and forms another phase. The other phases are very different, obviously. We want something like B here, a cloud emulsion, not such an emulsion. And yeah, I skip that. Here is one time more an example where microfluidics are better than batch. Yeah? So we get smaller nanoemulsion droplet size, Planck, and also when loaded with drug. And we looked also for stability. We made a microgravity experiment, and it was stable. Only if we torture the emulsions in the oven at 50 degrees, they decompose. I'm very happy. This was 4 days ago in northern Sweden, in Lapland, a sounding rocket launch, 309 kilometer high, 7 minutes microgravity. I forgot to mention, great thanks to German Space Agency, DLR. My samples were sent to space. This is Jens Lauslager. This is a rocket in the German colors where the samples were blown up to space, and my student is now analyzing it. I show you a second space experiment at the end. What we want to see is we coated the emulsions by chitosan to have controlled drug release, and you see it's controlled. The drug is released too fast, so we have to tell it release a bit slower, so we put a barrier. And this barrier works also perfect on our microgravity machine. But now we want to know, does it do in real on the rocket? We will know in 2 weeks when the experiments are done. This is a our microgravity machine. You see the nanoemulsions over there. It's made by Airbus Technologies. It's a random positioning machine. It goes all kind of ways, accelerates. This is a computer program, and people believe this is something like microgravity. Now we checked a bit deeper. There are different modes. I don't want to go too much into it. The RPM mode, random positioning mode here. The clinostat mode, this is like a spinning wheel, different speeds. You see the response is different, but generally it's clear we produce microgravity. Yes? This is 10 minus 5 G, 10 minus 5 gravity, and then it's a bit vibrating. But okay, we have a quite deep look into it now. This is a bit engineering. Question was, does the machine produce shear, and does the machine force coalescence of the droplets? The answer is no. We looked at it from the question of gravity, and we used the Bodenstein number. It would be critical if we could go here, but we are in a safe fashion with the Bodenstein number. We looked from the chip's free energy when droplets are supposed to coalesce. Everything is under it. The clinostat is a bit closer, so that is something you have to take care. The random positioning machine is in very good hands. My student Svenja, she's very good in making graphics. She summarized this in the paper here. So you see we are with the random positioning machine in the laminar regime, and with the clinostat, yeah, this is clinostat, we are a bit more in the transition regime. We have higher shear forces with the clinostat, but again, nothing is really critical. So what you learn is use microfluidics and make nanoparticles when you go to space, then you are best suited. And this is also shown here. This is a plot of dimensionless numbers. This is a Kratz number. This is the Richardson number, Grashoff number, sorry. This is the Richardson number. This is a Bodenstein number. We see under most operations, we are in the green field only with the clinostat. At forced convection, we can be outside. So we have a good idea how valid our space experiments are. Now I come to the last part. It will not take me more than 2-3 minutes. We want to make a tablet out of lunar material, and this is published. It just came out 2 weeks ago. And there are analogs, yeah? So, you know, we have to use excipients, all those materials, and that can be recruited from moon materials, as we showed here. And we made, too fast, we made a tablet coating by iron oxide, yeah? The red one is iron oxide. Iron oxide can sheet alpha rays. It's high atomic number. We cannot use uranium, obviously. We cannot use lead. So iron was the highest which we could use. So this is a shield against cosmic ray. Question was, does this shield work? That was a question of the experiment here. Now, it's fascinating when you do space experiments. We sat there, the rocket was ready, and 50 seconds before start, it was stopped. We had the champagne, and we all could go home. Okay, then you come next. So there are no launches in space. Yeah, I never say this. Only launch attempts. Yeah, and launches looking back. And finally, they made the launch, and we could enjoy our champagne. And thanks to Space Tango, and this is the University of Adelaide, the mission pad, and we made a second launch, all with Northrop Grumman, so not with SpaceX, NG-40, NG-15. And these are photos of our sample in space with an American astronaut, and this is our samples were outside ISS, yeah? Inside, here, and outside. This is a MISSE platform outside. And I got every time nice space experiments. This is all around our experiment. This is when they bring the sample outside. And by the way, our experiment is the first companion experiment, meaning the first ever inside-outside. Nobody did it before. People did outside, people did inside, but not together. Now, finally, we could evaluate something. We made here, I also claim we made the first systematic study. There were studies on tablets, but none of them was systematic. They used commercial tablets. We made a systematic study of the influence of iron oxide, excipients, and so on. And we saw inside ISS, tablets are safe. They also age on Earth. Ibuprofen decomposes quite fastly. Yeah? If you let it stand 1 year at home, you will have 20% degradation or 15% whatsoever. So this is what we see also here in space, marked here by descriptors, yellow and green. But if you go outside, everything is red with the exception of the iron oxide And to our surprise, flavor, terpenes, radical scavengers work. So a nice smelling tablets, and I mean this half series, is really good in space for stability. So that was a surprise. That is something we hoped for. And this is all documented here by UHPLC spectra and by LC-MS spectra, where we also analyzed a bit the decomposition products. Finally, what you could make with microfluidics, something I want to go for is very big ambition, look for the origin of life, to repeat the Urey-Miller experiment, if you're familiar with that. So that was 53. People still publish in Nature every year with that experiment, 70 years after the formation of molecules. The next step, Urey-Miller number 2, you could say, is bringing those molecules to polymers together, so proteins and DNA, with the help of nanoclays that has been supposed. And for some reasons, I think it's good to make that in flow chemistry because it's microgravity and because we need continuous extraction. I like to end with a quote of John F. Kennedy that he chose the moon and the space because he wants to make complex things which are hard, and that's, I think, what we all do in flow chemistry. If it would be easy, we would not like to do it. Okay, thank you.
It's very interesting, and there is some of the future topics, right? So do you know the ACS is have some space chemistry sessions every cases, right? They organize it. Okay, do you have some questions?
Yes. What's the strategy for just water and oxygen? Is flow chemistry, something that can be used for water and oxygen, or?
What NASA does with water and oxygen is something close to what we do on Earth with electrolyzers. I think that is the most reasonable approach. You might do it in a continuous fashion for some reasons. I cannot immediately say what flow chemistry could be good for it. Let me give another example, splitting CO2 from Mars into oxygen. That is what the NASA MOXIE system, M for Mars, yeah? And Ox for oxygen. That works very well, but Annemie Bogarts, who is one of the best in plasma, which I also do, made a paper, and she could show she's 100 times higher, or at least 10 times higher than the MOXIE system. So plasma, in this sense, could give a valuable addition. And when the chief of the NASA's Mars expedition visited my labs, he said they have a plasma team. I was really surprised because flow chemistry is industrial, but plasma is not really industrial, with a few exemptions. So I was surprised NASA considers plasma. But the point is we have to do more flow chemistry in space. Hardly anybody has done it so far. Yeah. Yes?
This is very interesting talks, and I think I can listen only here today, and not other place or other seminar. I'm very interested to know what kind of stability that droplet produce in microgravity or in the space.
Yes. When we talk about formulations and droplets, our calculations, which are based on validated engineering calculations on Earth at least, show that there should be not a problem with stability from terms of microgravity. The cosmic ray can be a problem because it can decompose like a coating, which we commonly have in those nanoemulsions. That was now the 7 minutes experiment. We will see what we get out of it. The most common effect is on the medicines, because medicines are labile, as we all know. They are labile on Earth. That's why we cannot use the medicines forever. And that is even stronger in space. What is missing is systematic studies. There's so much rumor in space. Yeah? Flavors are different. People say flavors are not different. People say medicines decompose fast. Not. That is like 300 years ago in science, and people get very emotional about it, like 300 years ago. We have to do more experiments and to bring more logic into the game. Yeah.
Okay, thank you.
Okay, so if you...
Basic question.
Yeah.
Very interesting, idea about this space chemistry, but I don't really catch the main motivation to focus on flow chemistry, especially in space resources. Like, is this because of the, let's say, the liquid, bulk liquid is not working as in this like Earth environment, or the footprint of this system, like manufacturing system, is smaller? What is the main motivation for that?
The footprint is a big argument on Moon and even more in a spacecraft. Like on a tanker, when you want to have a chemical plant, every millimeter counts. Every millimeter is millions on a tanker when you have chemical plants. But again, flow chemistry is the only technology which works under microgravity. Yeah? All other technologies work less or partly. Flow chemistry is on Earth under microgravity. It doesn't matter if your flow is like this or if it's like this. Yeah? Because with the gravity problem, I never thought about this. Yeah? They're usually very chaotically arranged. Yeah? Because it doesn't matter. Yeah? So that's a big argument. But I'm not saying all in space will be done by flow chemistry, but it has some ideal features and process intensification, fast, high productivity, personalized process, from Peter Seeberger, process automation, all those things which we love about flow chemistry. This is what we do. When you have a moon village with 20 people, you cannot have a chemical engineer, a policeman, a nurse. That will not work. Yeah,
so automation or controllability is the main motivation.
Once we have 10,000 people on Moon, I would say it's no problem anymore. But when we have 100 people on Moon, it is a problem, like on the small. So we need highly automated system. And we cannot bring by payloads. That is at least what all experts say.
We have to contact Elon Musk, who is going to be build up the habitat in the Mars.
Yes, yes.
Do you have any question?
Thank you. Is there any distinct or critical difference on the crop cultivation on the Moon and between on the Moon and on the Mars?
Again, there are a lot of rumors, and the literature is not systematic. Some people say the roots go more diverse, they don't go as straight as on Earth. Other people say roots follow also light, so you don't need gravity for a good root development. It probably depends on the plant species. On Earth, everything is a bit compressed, also my cells here. Yeah? So people have shown that on space, when you look on lab on a ship or organ on a ship, you have the better, what they call physiological conditions. So there are probably a lot of differences, but investigations are not so big to tell all the information. We have not a lot of, but we have some failures in the NASA system, other systems. Yeah, there's a dispensing system for water didn't work for a little while. What he has shown, in his honestly in his lecture. So and then they really struggle very hard to get the tomatoes alive in space, and they have 2 or 3 days no water or no light. Yeah, so that happens. That is reported. Yeah. So process control is also a problem. Sorry, I'm not a plant biologist, so I do the engineering around it. The plant biologist could answer you probably better. There is something different, that's for sure. But the question is to what degree is that?
Maybe at this stage in the flow chemistry, chemists may have a chance to opportunity, like participate in some program of Mango or like Alpha, and there is under some microcavity condition, and there is a satellite stations, right? And there is a day for sometimes recruit some or call for the programs. Right? So there is maybe any like some of the researcher in there, no matter what any nature, so we can participate.
I think flow chemistry has a big chance in the transformation of regolith into valuable materials, so minerals processing we would call that on Earth. it has a big chance in the extraction of bioactives, like beta-carotene, all those health nutrients from plants, and a big chance, what we have seen in the first talk, on the formulation of nanomedicines. As I've said, actually there's more liquid. I was surprised. I think they take only tablets because it's easy, but they take half of what they take is liquid formulation, so there's a big need in space for that.
All right, so he's going to be give us another chance to give a talk in Busan, so we can make a longer discussion. Right? Thank you for this. Thank you. Coffee break session, and then it's something. Yeah, let's have us about 13 minutes, make a coffee break, and then please come back at 3:50. Right? 3:50.
Unfortunately. Okay, so is the next speaker, and that is from professor from Taiwan. Actually, he's some of the Japanese Koreans, right? Right. So his name is William Liu, and that is Professor Lee is the American Korean. So I'm a Korean. So. And actually, he retired and then Osaka and the Prefecture University. Now it's Osaka Prefecture University. Changed the names, right?
Yes, merged with Osaka City University to
Osaka.
make Osaka Metropolitan University.
Metropolitan University right now, right? It's combined together with the others, right? So then I think now he moved to the Taiwan, and then still he's doing the teaching and the researching, right? So he is really one of the long career in the flow chemistry society. Okay, thank you. Welcome to the speaker again.
Thank you very much. Yeah, thank you very much, Professor Don Kim. So always happy to be here. So actually, I was born in Japan, but I'm Korean. But don't trust my Korean language, so don't test me. So this today's my topic is very basic chemical reactions using flow chemistry. So unfortunately, I cannot show the space picture or something. Okay, so this is our world. Our world should have a chemical industries, but chemical industries should be compact in this era. So they are changing. So something like this, we should have a compact flow plant that we can, how do you say, survive, that chemicals can survive with the harmony of this our world. So in this regard, flow chemistry is very important. I took this picture, this scheme from this perspective continuous flow chemistry that published in some years ago in OPRD. And of course, everybody knows the advantage. It's faster, it's safer, and we can challenge several reactions with flow chemistry. So that is not available in the batch chemistry. And probably product quality should be better. And the important thing is smaller footprint. So these are the advantages of continuous flow chemistry. And actually, if you are interested in the very basic flow chemistry, to visit this video. It's a silo in Melbourne, so they have a very nice animation. So it's just 5 minutes. I highly recommend it. Okay, so this slide shows that this IUPAC always do the things that they are selecting 10 chemical innovations. So this is taken from the 2019 IUPACs. So 10 chemical innovations that will change our world. So that is flow chemistry here. So already organic catalysis, 2021 Nobel Prize got there. And very recently, interesting mechanochemical techniques also appeared. Some others are here. And so flow chemistry is now, people always pay attention to the development. So I always use this slide for the student to show the concept of flow chemistry. If you pour 1 milliliter of the liquid to 1 square meter of this, something like this cubic type of we pour here, the whole area is 4 square centimeter, just 4 square centimeter. But if we use a long, okay, here 100 micrometers, this one, the length becomes 100 meters, whole area becomes 400 square meters. That has a meaning of this diffusion, excellent diffusion. Through the wall, we can take the heat quite quickly. We can give a heat through the wall very efficiently. And the other thing is, if you think about A and B react to give C, in case A and B have to swim to see each other long way. But here, quickly they are mixing. These are the great advantage of microspace reactions. And I will show you very basic example. Heat management is very easy. So what we did is, actually I have been long been involved in radical chemistry, so I'm interested in the radical thin hydride mediated radical chemistry that can give this reduced product of organic halides. So typically we use a thin hydride. So if you check papers that reporting these thin hydride-based chemistry, always they use thin hydride stoichiometric command and AIBN as a radical initiator, and heating 80-degree, and reaction time is, some paper reports 3 hours. Refluxing, benzene refluxing 3 hours. Some paper 6 hours. Some paper overnight. So then I thought, so if we apply this very simple, very basic radical reactions, radical reduction to flow system, that should be, reaction time should be condensed because of the thermal efficiency is great. So then I asked student to try the reactions. So then he reported me 74%. It's 1 minute. 1 minute, 74%. Not so bad. Not so bad. But he also reported me that significant amount of AIBNs remained unreacted in 1 minute. This is very reasonable if we see the half-life times of AIBN, that is at 80 degrees, 2 hours. So that is why people use the AIBN. Why people like AIBN? It's a safe radical initiator and human being friendly, very easy to use for us to run the batch reactions. But in turn, if this special radical initiator is V70 or V65, that decomposes very quickly. So half-life times at 60 degrees, this guy's just 10 minutes. This guy, 12 minutes or something. So that we have to keep them in the refrigerator to store, otherwise it's decomposed, self-decomposition takes place. So then we asked students to try this in place of AIBN. Then we got almost quantitative conversion is obtained within 1 minute. So we published a paper, Organic Letters, and we see this Peter's papers. So he also used a very nice system. It's a xanthate reduction that using TTMSS. This is silicon hydride. It's a thrower reagent, but it's a safe reagent because tin is notorious for the toxicity. So we are surprised to see your paper at the time. Anyway, so then we have a long collaboration with Professor Dompyo Kim. So at that time, he has a PI, polyimide reactors. That is, for me, very strange reactors moving, which very flexible. So we tried to use that one. And also it worked, but there's some radical reaction. It was good to have a joint paper in Angewandte Chemie. Okay, so this is some work with collaboration with Amidou Studa in Münster. He's a long friend of mine. So for this compound, it's a tempo tempo-attached malonate esters that undergoes homolysis when heating. Homolysis gives two radical species. One is transient radical species, it's a reactive species. The other is very stable, persistent radical. It's a tempo. It's commercially available. It's bottled and you can buy, purchase for this. So then amidostuta was involved in this alkene addition of tempo malonate. So malonyl radical add to this terminal carbon, then tempo comes here. This is a desired product. But he suffered from this in the conventional batch reactors. So this is 39%. This byproduct is formed. Obviously, reaction takes place. This should be because of the further reaction, the carboxylation reaction, so lower the yield of the desired product. So he used microwave heating, quick heating. So then he improved the yield. Still, he got the byproduct. So then we tried a microflow system. Then we were able to get this as a sole product. So only trace amount of this. Why? So this is because after heating 180 degrees, it takes time to cool down in a batch system. So this is because in flow system, quick heating is possible and quick quenching, quick cool down is also possible. That is very good. That's why we prevent the formation of byproduct. Okay, so we are long interest involved in the flow photoreactions because of this Beer-Lambert rule. So thinner is better. So everybody recognize this. But we are proud that we are one of the first people to be involved in this system. And so far we have been in the many devices. One time we paid a lot of money to Germany to buy expensive microglass one. Anyway, so we are fortunate to have an opportunity to summarize our historical contribution to this area, recent journals. And I will tell about a little bit why I have been involved in flow photochemistry. So this is a Barton reaction. Professor Barton is a Nobel laureate. And he has so many Barton reactions, famous name reactions. But among them, the Barton nitrite reaction is what we like. The reaction is starting from nitrite, and the photolysis gives a radical pair. Again, this is a transient radical. This is a persistent radical. They can stay, nitroxide. So then 1,5 hydrogen shift takes place to form radical here. In the end, this nitroxide combines with this to form nitroso compound, but its enol form gives the oxygen as a final product. So this is Barton reactions. And my friend at Shionoi Pharma in Osaka one time asked me, actually Dr. Konoike, so came to me, so they are interested in this compound. This compound is a precursor to the mycelic acid that has an effect of hypotensive effect. And the problem is if they have more amount, so this is a very obstacle to spend a lot of money for huge light source. So then we started a collaboration with him, his group, and then we noticed what wavelength is necessary to cleave that ON bond. That is around 360. So this is these are not necessary for mercury lamp. So that's why we use filters. That causes energy, huge energy loss. So then we finally found out this 360, it's maybe this very cheap Toshiba's black light is enough to do. So then we switch to the, something like this black light, Toshiba's one. And this is a microglass at the times. So we use this, and then we try the reactions. And then this we try to scaling up using this long flow reactors and using 4 pieces of 20-watt black light, so 8. Then what we did this, and we got this desired product. But after running 20 hours, just only 3.1 gram. It's a little bit discouraging. So that was so discouraging. So then new idea comes. Reactor is compact. Compact reactor can fit maybe compact light source. That's our idea. So downsizing the photoirradiation source. But we thought, what about the use of unbalanced, very powerful light source to compact reactor? That may be something new. So then, actually, Kyocera is a company, Kyoto's company, so that has a very nice LED systems for other purpose. So I connected with a technician at Kyocera, and we invented a new type of UV LED that has 60 to 600. So this is, I think, in our opinion, according to Kyocera, this is the world #1 powerful LED line. So it's very useful. So we try. Okay. So at the same time, actually, Mix is our spin-out company. So then they made this series of photoreactor that is mechanical carving or photo etching, and then they pressed with a glass plate, and very good one, very heavy one. So we use this for the flow reactor. So then I will show you 1 or 2 examples of this area. So we were interested in using molecular oxygen. Molecular oxygen is abundant, very cheap, of course, but sometimes it's dangerous when scaling up. Peroxide can produce a lot, so that is danger. So people don't like the scaling up of the molecular oxygen reactions. So in another project, so we are involved in the TBAT chemistry, decatungstate anion chemistry. That is a very good photocatalyst, very cheap. And then this photo-excited species can abstract hydrogen from alkyl RH bond to form radical species. So we are involved in this chemistry. So then we encounter this catalyst is used for the oxidation of CH oxidation. So then we try to use this UV LED, strong UV LED for this using oxygen gas. And benzylic alcohol was mixed, and then we were able to get desired benzoic acid 71%. So this residence time is just 5 minutes. If we run the same reaction using a batch style flask, that maybe a couple of hours are necessary to finish. So that is, we were able to speed up the reactions. So then this is another example of this molecular oxygen-induced CC bond cleavage using the same catalyst, photocatalyst. So then we were able to get these carbonyl compound very easily, but it takes, see here, 12 to 18 hours generally. So then, of course, we are motivated to use our system here. So this is a blue color is a solution of this decatungstate anions color. And then this segmented flow is between the blue one, there is a oxygen here. So this is channels. So then you will see gas part is diminished in the end. So that is reacted with, so it's absorbed. So in the end, no gas. So actually this was, oh, it works. Okay. Okay. So this, in this way, so maybe we can, how to say, we can think about the use of molecular oxygen. It's supposed to be thought as a very dangerous reagent for the safely in the industrial process. Okay, so then another topic is chlorination. Chlorine is so dangerous, much more dangerous. So nobody wants to use chlorine gas. So there is a, so we thought sodium hypochlorite mixed with HCl. This is a textbook chemistry of a high school student textbook that give you chlorine gas. So then we reacted with alkanes, and then after the reaction goes, toxic chlorine gas disappeared to get the product. That's the basic ideas. So, but Oliver Capet scooped us, so then we are obliged to publish the same content to the same journals. So that's okay. So then this one time, so when I have a lecture for industrial people, they complained about that. Why use this one? You should use chlorine gas. So actually, chlorine gas is so cheap, okay? Maybe it is 400 won per kilogram here. So cheap because we have to produce sodium hydroxide from this salt. So this is a byproduct. So soda industry always have chlorine gas. Very cheap. So then we decide to use one. So then if we see the history of this flow chlorination using chlorine gas, there is a paper from the IMM, it's very famous. These foreign film reactors, so expensive. We can't afford that. We couldn't afford that. But anyway, so this is a work that gives benzylic chlorination. That see here, residence time is 40 seconds, just 40 seconds, very fast. But we saw the productivity is using these foreign film reactors 1.65 grams per hour. It's a good to show that is to show the principle of the usefulness of this reactor, but it's still far from the industrial applications. So that motivated us to prepare new reactors, photoflow reactor. Its length is 60 centimeters. So what we did is we thought we use it's like this. There is a slope, and the substrate is coming here. Then because of the gravity control, they go through this, and chlorine gas is introduced here, upper side, then photoirradiation that gives chlorinated product here, and then the byproduct HCl is here from this. So we decided to test this one, and of course we use the high power UV LEDs for the photoirradiation. First, my student tried benzylic toluene's chlorination so fast, 10 seconds. 10 seconds already double, triple. So fast. So it's so even though this reactor is used, maybe if we count maybe 200 grams per 1 hour. So then, so some company in Japan, so near Osaka, hope to prepare this one. They have actually chlorine gas. So they come to us and for the chlorination with ethylene carbonate. This is very cheap. So then the chlorine, then the base gives this vinylene carbonate. this is very useful compound. So they are interested in the workflow system. So this is a byproduct. So second chlorination always happens. So then we try this reaction and using this, so very scary since we have to use chlorine tank. So, but, okay, so first we use a mass flow controller. Mass flow controller, once we used, maybe after 1 hour it stopped. Anyway, so we cannot use mass flow controller. So no mass flow controller, with analog float meter we used. But it works quite nicely. So it's 30-second residence time, it's okay. So then it can produce 8.5 kilogram per day with this 60 centimeters one. So if people increase one, so it's quite easy to, to industrialize this cloning process, we believe. So this is something like the photo of this one. So irradiation is take place. We checked which place is good for the irradiation. But actually this is a radical, irradiation is necessary for radical initiation. It doesn't matter. So just some places, that's okay. So then this reaction is applied to the NBS, flow CH bromation, NBS, benzylic one. So that is 6 seconds, very fast for this. And this brominated compound is obtained using this, our equipment. So this is 6 seconds, but if we see previous work, there are 2 works we found. It's again Oliver Cappe's work. Benzylic bromination, they use, 47 minutes. 47 minutes. Well, 6 seconds. They use this type of and also Horn Christian, so Melbourne. So he also used Corning's photoreactors. Again, we checked 10 minutes. So our reactor is the best at this stage. Okay, so some other application is synthesis of pristine. This is obtained from the basking shark, but we cannot take basking shark anymore because of Washington treatment. So we have to prepare by ourselves. So Koichi Fukase published a couple of years ago. He succeeded in preparing pristine. So using this triene as the starting materials, and then paratoluen sulfonic acid. This is acid. What he did here is to eliminate OH to form double bond, dehydration. So then after that, the product is submitted to, it's a batch, plasmon carbon catalyzed hydrogenation to get pristine. It's a nice system. But the big problem here is the use of paratoluen sulfonic acid. So their protocol uses a stoichiometric amount of paratoluen sulfonic acid. That if they prepare pristine by this route, they suffer from the huge amount of waste, acid waste. So we thought this can be catalyzed by the different way. So this is we like this catalyst, silica gel supported sulfonic acid. So this we call HO-SAS, since especially HO is here. So then we try this, and we mix the solvent, mix the catalyst, 10% plasmon carbon. So we tried it with just this unit can contain both catalysts and then flow with hydrogen starting from that same alcohol. Then we got this pristine in its 30 seconds of this very quick reactions. And because tranexamic acid is used for this some so that is so many purposes. And actually, I was surprised since maybe 6 months ago, I got some medicines from this my hospital. So that was tranexamic acid. So that is, I can prove that is commercially market. They have a market. Yeah. So then to make this one, there is an interesting paper in OPRD. It's a batch system, but their system is starting from cyano benzoic acid esters. So this have to be reduced. Cyano group have to be reduced to amino group, and this benzene group have to be reduced to the cyclohexane. So actually, so my colleague, Professor Sato, already published a paper about this. He started with cyano carbon compound. That is, when they use a batch reactor, no cyano reduction, but in the flow system using a packed bed reactors, he got significant amount of cyano reduced products. So that means flow reduction is maybe efficient, much more efficient. So we checked the several conditions. Finally, we were able to get one flow synthesis of tranexamic acid. Okay, here, starting materials. First, we tried paradimon carbon reduction of nitrogen group to get amino group. Then this amino group, so after that, we neutralize this. Then we submit this to this ruthenium alumina complex. So hydrogen is always coming through here. Then we are able to get almost 100% yield of the desired product. So this, we obtain trans and cis mixtures. The real one we need is maybe trans. So we need some treatment with heat treatment. We are able to get this 87 to 13 mixture of this one. So we need separation of this, but this is a very convenient route from starting from the compound and just only using one flow systems. And then so now we are interested in the ultra minimum reaction space. So far we are involved in this flow chemistry, microflow chemistry. Then our idea is how to scale up to accommodate the productions. But as you see, microflow system has a huge advantage in this ideal reaction space. If the space is bigger, so many problems of the diffusions. So that's why so many process chemists are working in the chemical company for the scale up. So we thought we should do this way, not this way, this way. So then, even a micro batch, that should be very ideal for the reaction. So, but we cannot make batch flask, such a ultra minimum batch flask. We cannot have any idea about that. So then we came to the micro droplet. This should be a reaction apparatus. That should be very good. And then our idea is this micro batch reactor should be very good in terms of surface and volume ratios. And then if we got information about that, okay, reaction information using one droplet, we should jump up to this here. So that should be very fast, very quick for the production. So that is our ideas. And our idea is droplet moving the phase cause a vortex mixing. If we never move that very slow, very slow. Mixing is not expected. But moving the phase, that can be mixing. So here is a micro droplet that's continuously generated, and we are planning this. And also the other idea is if we number in the first droplet, second one, so then if we give a different energies, we can get information quickly. So that's the idea. So droplet was segment that is a part of the big flow system. So we should get information with this cutting. We cut out this part and then know what's going on. So then our idea is firstly this one, gravity control droplet. So this is a dehydration reaction to get agrochemical target. So first, this is going up. Should be this one. Then on the top, the second one is coming, chasing the first droplet. And finally, it mixed. Then reaction takes place. So in these reactions, we use 3 milligrams of starting material that is 20 micromole. And then we check that treating this at the room temperature for 3 minutes gave us 81% of the product. So you may see why this is going, this top side never going down, because this we use some heavy solvent for this. So this cannot go this. So actually this heavy solvent is moving. So, but it's okay if we fell down that system that we can easily take this part. Okay, so this is a gravity controlled one. So then we compared with some other reactions, single droplet reactions, 10 droplet reactions, 10 continuous droplet reactions, and completely flow reactions. So yield are quite similar. So that means we may predict the real to ideal flow reactors condition, continuous flow reaction conditions by just checking one droplet reactions. So we are now expanding this idea for some other reactions now. Okay, so this is our concept, high-speed information for continuous flow conditions. User thinking so far is information to get microflow, then some scaling up for this ordinary flow system, pilot flow, then flow production. This is an ordinary way, but we think we should jump up from the ultra-micro batch reaction to get information on flow reactions. That's our ideas ongoing. And actually, this is our new laboratory. So our spin-out company has a very new lab in the Amagasaki area. Amagasaki is next to Osaka. Very good places. Actually, there was a laboratory of BASF laboratories. Yeah, they left. So there is a vacancy. So we were fortunately entered the place. And this flow project was mainly done by these 5 doctors, PhD degree holders. So I appreciate this contribution to these coworkers. And also I like to appreciate along my coworkers at Osaka, Professor Sato and Professor Fukuyama. So, and also funding from the JSPS and NIDO is highly appreciated on this opportunity. Okay, this is in our Amagasaki's laboratory, so this is our recent pilot plant. So this DCBT means dicyclopentadienes, some molecules to give cyclopentadienes. So that was done in this area, and then that's home to cyclopentadienes fed into this second flow reactors. Then we mix with dianophyte to get this product. So then we tested 3 months. So then we are able to get 1.2 tons. That is the first experience for university people to be involved in some industrial production. That was, we learned a lot anyway. So that is because of the researcher don't want to stay long at night. So we start from 10 o'clock to 5 o'clock every day. So then, of course, we don't operate this plant for this weekend. So then it took 3 months, or 3 months. But anyway, we are confident now we can be involved in the chemical production. Okay, thank you very much for your kind attention. So this is in Switzerland. I took this photo. So many flow, and to get here ponds, and the people are living with that water. Thank you very much.
Okay, thank you. Always I admire his chemistry. So any question?
Yeah, the last part of your experiment about the GCPDA analysis.
Yes.
Usually we are doing the distillation to separate, and then during the microbe or flow reaction, it comes out only one product out, or you did the separations of that.
So I didn't bring the photos, the precise photos. Maybe I'll find it in the Busan.
Okay.
But anyway, so that we have a first. Not just a simple distillation cannot give effectively the product. We need some time for the decomposition. So we use some oil pool. Then the heated dicyclopentadienes drop down. So then it's becomes gas, and then we use distillation.
So some kind of the
separation
by distillation.
That is, so.
Any further questions?
Very interesting talk. Thank you. Because I am a chemist, focus on the flow chemistry process development, and I'm very sorry I cannot go to Busan tomorrow because of my business trip.
You missed very important talks.
Yeah, I wanted to know more detail of your ultra micro batch strategy.
Yes.
Yeah, and direct to go to flow production. So could you give us the more detail about the strategy? Yeah.
Okay, so just I show this is a one-drop systems. But actually, this is not so practical that I show you. It's for this exhibition. So we actually use a continuous droplet reactions like this here. Yeah. So this is easier to make droplet continuously. It's easier. So then the reaction is done. The reason why we use this one is we combined with inline NMR and inline IR to know what's going on. So especially NMR requires some amounts. So what we did is this after the reaction, this is accumulated to make a bigger segment. Then we separate. Separation is actually we use Garden. Garden is a perfluorinated polyethylene type. It has a density is 1.7. So it's go to the earth, yes, center of the earth. So then this organic part is floated. Then that move to enter this NMR flow cell. Then we can monitor that. Yeah, so that is, so then the, actually, this continuous, okay, so droplet flow is still necessary. This is ideal. Just one droplet is just ideal. So this is more practical, and we have some other reactions. Okay, so like this one droplet, 10 droplet, anyway, continuous droplet and flow is quite similar. So correlation, we confirmed this using some other model reactions, maybe 3 reactions, all well. So then we think we can, how do you say, just spend a short period of time, we can know the answer of this flow production. That's our idea. Yeah, maybe you can, if you can come Busan.
So that and then you are going to come to actually, she is has some like opened her own startup.
Ah, really?
Really chemistry.
Very small.
She's very
inviting.
She's so busy, and so
Okay.
she could come to the same.
Please invite me.
Okay.
I have a question to this. I'm following the reactor.
Uh-huh.
What you built is also a falling film reactor, yeah?
Mhm.
So that's nothing different,
Mhm.
but it has a different angle.
Yeah, yes.
I know the INM reactor can already operate below 100 micrometers, very thin. Yeah? So is the reason you have 10 or 100 times more productivity
Mhm.
because you have stable, thicker films? I would
Mhm.
say that is probably the reason that you have more productivity.
Okay, so.
And that would mean thicker is better than thinner, which is.
Oh, yes,
against
yes,
our
yes.
intuition.
Yes. So actually, falling film reactors, it's maybe the similar. So our idea that was similar. Yes, but that cannot be, how do you say, good for this more, how do you say, to get more product, right? Just so then we saw that the good thing of our reactor is all are made of glass made. No contact of the chlorine to this. Just we use a metal frame. That's a frame. So that is, and also this. Okay, I cannot explain well, but that is the. Anyway, that is has a 10 centimeter wise. So that we can make more and more. So that is, even we can make a ton scales. But I'm doubt the falling film reactors cannot do like accommodate to this scaling up, right?
We built larger falling films.
Larger?
Larger areas. But I would say you use thicker films because you can over-engineer. Germans are known for over-engineering, so probably you make thicker films, which are not so easily to stable, but because you have 15 degrees.
15.
Yes. It's not stable like this.
Yes. So
that's probably a great idea.
Yeah, thank you.
Okay. If not, and then it's also some thanks to speaker King. So we have like a last speaker. Actually, I'm missing the one speaker has not been arrived, and there is from Australia, and there is a University of Melbourne, and there is Anastasios Polizo. He's another like organic chemist. So now he's coming. So he is going to join us at the dinner times. So, okay. So and there is some of the Peter from Max Planck in Portugal. And actually, he told me that he gave a talk this morning in Hangzhou, 9 a.m. then fly to Minchon, and then he provide his 2 talks in a day, right? How efficient, or how crazy you are. Okay, so this is something that he's going to give now he is going to be one of the, quite challenging program. He is leading. The name is the Center of Transformation Chemistry. The transformation chemistry means is some of a completely recycled chemistry, not just from some of the petroleum-based chemistry. He tried to make it some of the new organic chemistry products. He tried to build kind of. So tomorrow I arranged some meeting with our university president, because our university president is also a chemist. So I met them together, and I hope that my university president got inspiration from his program.
Okay, let's hope so. Well, first of all, thank you very much for the invitation to come to Seoul to give this talk, and look forward, of course, also to Busan, where we see most of you. You asked us to give more of a biographical sketch, so I thought I'd put a few things together that we work on over many years, and what is sort of a recurring feature for our research is the fact that I'm interested in biomolecules, and so should be most of you, because biomolecules make all of our life possible. Nucleic acids store information, transfer information in the form of RNA. They carry out basically all of the work in our bodies in form of proteins that are the workhorses of biology. And lastly, polysaccharides that all of you eat typically are considered the dark matter of biology, because they really are construction materials. The reason I bring this up is that a physicist, Richard Feynman, once famously said that I cannot create, I do not understand. Interesting enough, it was actually on his board up here, and this was the blackboard in his office the day he died. I don't think he died because he wrote it on a blackboard, it just happened to be there at that time still. But I think we can help a man, because as chemists we can actually create, and we already saw today the creation of chemicals in all sorts of ways, and that is the common feature throughout my talk today. What I like to point to is some very simple chemistry, and that is the chemistry of making DNA. Now, if you think about it, that is a very, very simple reaction, seemingly for organic chemists, where nucleophile attacks a phosphate monoester to make a phosphate diester. You have 4 building blocks, you have 1 chemical reaction, there's no new stereogenic center that's being formed in here. So how hard can that be? Well, it turned out to be actually pretty hard. In 1976, it took a 29-person team to make a gene, 126-mer. Earlier, Corona had made another gene. It's about the same thing. But the interesting part here is 17 consecutive papers. So if you think about it, totally crazy, right? Nobody's going to repeat it. Who's going to do that? Well, my PhD advisor, as it turns out, he developed automated DNA synthesis in 1981, and that really ruined the field for most people, because, and I just use this now as an example, in 1998, 1 2-author paper synthesized 123 and 126-mer, and the synthesis was a footnote. And this is what many chemists are afraid of. If you can do the chemistry really well and you can automate it, you make the chemistry a footnote. But I think that is a misconception, because there is still nucleic acid chemistry to this day, and of course this has really enabled an entire field. Now, I should also tell you that most of my students believe that it's DHL that actually makes DNA, because when we send an email, 2 days later, DHL envelope comes with a DNA in it, okay? It's, of course, made other places. Okay, so we understand one reaction. We can then think about automation. This is applied biosystems synthesizer, started also by Carruthers. Then you have Emerald Cloud Labs, where DNA and RNA is made today, totally automated. There's no more human intervention in this case. And then, of course, you can have commercial utilization, such as companies such as Amgen. So my PhD advisor started both Applied Biosystems and Amgen. He became a multi-billionaire through that. And what I realized at that time already, if you do good chemistry, you can do good biochemistry, you can do interesting medicine, and you can make a lot of money. And all these things I find very interesting. So it turns out, of course, many other things have been enabled by DNA chemistry. And to me, from a chemistry standpoint, But one person can make DNA and RNA, enable basically an entire area. And all these things on the right side have one thing in common. They all got Nobel Prizes. And they have something else in common. None of these things would have been possible without the synthesis of DNA. The synthesis of DNA or RNA never got a Nobel Prize, which I think is a total travesty because that totally enabled all these other areas. These other things would not exist. Of course, the Nobel Prize was given for the idea of solid phase synthesis because Merrifield in 1963 showed us how to make peptides. Again, instruments came about, peptide arrays, peptide drugs, protein engineering, and of course, this past year, now this year, there's actually AlphaFold, which they got a Nobel Prize for it. But I would again argue, without being able to make all the structures, it would have been very difficult to understand them. So for me, as a PhD student, I'm a pretty simple-minded kind of guy. There was peptide chemistry, there was DNA chemistry. Okay, DNA encodes peptides, peptides make carbohydrates. Nobody had done this. As a PhD student in 1993, I said, okay, I'm going to make automated polysaccharide chemistry. And then most people said, that's totally crazy. You're going to totally fail because that's impossible, because that's much, much harder to do. But let's get to that in a second. Why should we be interested in carbohydrates? Not for nutritional reasons now. I'm more interested in them because actually carbohydrates are the predominant biopolymer. So if you look at biopolymers by mass, it's by far carbohydrates, mainly in form of crab shells, cellulose that make these trees up. And what's interesting, I won't talk about it today, but that's a big part of my research program. Nowadays, we are very interested in algae. For every kilo of cellulose on the planet Earth, there's 1 kilo of seaweed. And the interesting part is we basically know nothing about the carbon cycle in the ocean. So in all the climate models, we only consider the stuff on the surface of the Earth, but none of it in the ocean. And that is actually pretty interesting because with global warming, it could mean that more carbon is being put in algae, or it could actually mean that more algae are being eaten and more CO2 is being liberated. We don't know that, and nobody knows it, and that's why my lab today works actually a lot on algae. Another area you're interested in is the human cell. Now your cells, for most part, are covered by this black thing. It's called the glycocalyx. So these are complex carbohydrates. This is an example from tuberculosis bacteria. And the carbohydrates on those cells are specific for particular kinds of cells. And lastly, very early in your life, you probably don't remember it anymore, but when a sperm cell gets together with an egg cell during fertilization, that is a carbohydrate-protein interaction. So carbohydrates are also important for signaling. Now, when we said we would automate carbohydrate chemistry, people said, okay, that's pretty crazy because in DNA, there is no new stereogenic center. When you form it, neither is it an amide linkage. It's glycosidic linkages, when you make it, where you have a new stereogenic center, and this oxygen go up, or it can go down. So that means you have to exercise stereo control. The second thing is, carbohydrates are oftentimes branched, which means you have to exercise retro control. And to make matters worse, we oftentimes conjugate it into proteins. 85% of human proteins are glycosylated, so you also have to make conjugates. And there are also modifications. And so that turned out to be relatively difficult. Now, what we did a long, long time ago, my third year as an assistant professor, we showed that this in principle can work. And again, we invented nothing new, right? We just used Merrifield's resin. We developed new linkers for that. And then we would wash with acid. Then we put a building block on top. The building block would be connected to solid support. We do that typically at low temperature. Then you do capping of any unreacted hydroxyl groups that should not react in the next step. And then you're going to do deprotection to liberate one hydroxyl group, ready it for, again, the next reaction. If you like, you can do functionalization with phosphates or sulfates, and then the machine just keeps going through that coupling cycle. Now, what I should also say, in this automated method here, this is about 2 years ago, we used to take 100 minutes for the incorporation of one sugar. Now, when I was a postdoc in Darashevsky's lab, 3 postdocs took 2 years to make a 5-mer. One of my students makes that same thing today in less than 5 hours. So today we are at approximately 45 minutes, and I think we are getting very close now to getting it down to about 20 minutes per building block. So we are shrinking this time as we speak. There are some details, maybe I talk to Busan about it, but that is a lot of reaction chemistry where we are thinking about, which for today I think goes too far. Afterwards, we remove the material from a solid support using photocleavage, actually in a flow device, in this case, global deprotection, and then we get the glycan in the end. And what it shows you is that with time, we went from very short things like 9 mers in a science paper, and the record now is 150 mer, but 100 mer in one go. Those are 201 chemical reactions, 100 couplings, 100 deprotections, and 1 removal from solid support. A Chinese group has since used our chemistry, went over 1,000 mers, but honestly, it is not so much of a thing who's got the biggest carbohydrate. In this case, it's really for us to show we can do it, but for most purposes, we don't need such large carbohydrates. Now, the other thing we're able to do is we started to think about, can we actually resolve the structure of carbohydrates? And that was very, very important because we don't really know in many cases what does the structure look like. Together with physicists at Max Planck, we looked at this SDM imaging, electron spray, mass time of flight mass spectrometer, land on a surface, flatten it, and then at low temperature use STM to actually look at the structure. And this was why it's in a big journal, because for the very first time somebody was able to actually resolve the structure of a single carbohydrate. You can then observe pretty cool things, like this molecule here looks like cyclodextrin, 6 glucoses, very nice. The only problem is this is not cyclodextrin. This is actually a linear structure. It's this synthetic molecule. The only bond that's missing from cyclodextrin, it's from here to here. And the interesting thing is in the gas phase, that is actually the lowest energy conformation. But then you take a tip on a surface and you hit it, then it snaps open, it becomes linear. Is it biologically relevant? No. Does it look pretty cool? Yes, that's why I show it to you, okay? But that's sort of the only reason. The good thing is we now, for the first time, can actually see single molecules in carbohydrates and look at them. Now, machines that we built, they used to be very large. This is the Glycine 2.1, the first version, built by some of my students that shipped that. It was shipped all around the world. For the last 2 years now, we built an instrument that's 4 times smaller, 4 times faster, 40% cheaper. When do things ever get cheaper, right? I mean, that's important as well. And the next systems, the next one is going to go, I think, in Singapore, one in China, and then one in Manchester. So the last year, they started selling a lot more instruments because they become much less expensive. And so I think that's why people are actually using these machines. Now, once you can make the molecules, the question is what are you going to do with molecules? And the bad news is, most likely we're all going to die. The question is, how are we going to die, right? Today, cancer is one of the predominant causes of death. Diabetes is a problem. And what we see are antibiotic-resistant bacteria. And if we believe the numbers biomedical doctors, in 2050, more than 50% of us, or our kids, could actually die from bacteria we can no longer treat. And that is a pretty scary thought. So for one, we have to make new antibiotics. But for second, we should think about, can we actually prevent infectious disease by making vaccines? Now, the reason I bring this up here is most bacteria are covered by a so-called capsule, and that capsule consists of polysaccharides. Now, I showed you earlier, this is the glycocalyx around your cell, and mammalian cells, if they would be donuts, would be pretty boring, because they only consist of 9 monosaccharides, and, well, not that interesting. Interestingly, cancer cells actually change the carbohydrates on the surface, which means I can create ways of detecting cancer cells, and I can take ways to actually kill cancer cells using either antibodies or vaccines. I started one company that right now is starting human clinical trials using antibodies we produced against carbohydrates on the surface of cancer cells. But for today's lecture, I'd like to focus on the crazy guys. This would be a donut if it would be a bacterium. Many monosaccharides, lots of diversity, and totally different carbohydrate structures. So the idea of vaccines based on carbohydrates is to make an immune response against these carbohydrates on the outside, very specifically, and kill bacteria. Now, if I look around the room, many of you will most likely have been vaccinated with carbohydrate-based vaccines. There are 3 vaccines that are used today. Haemophilus influenza type B is used in almost every country, except for our Japanese colleagues. You did not get it. In Korea, I don't know, but in Japan, they don't use it. This was introduced in the Western world, the United States, all of Europe, the rest of Asia in 1992. During human clinical trials in Japan, several children died. It's completely unknown to this day why that happened. It could have been something totally different, and that's why in Japan, it was actually never approved. Which is really a shame, because if your kids get that, this is really nasty. There's a severe chance of dying. 50% of the kids who get that die, and the other 50% can have very, very serious health issues. This one became very famous, is Prevnar. Today there are other things which are working against Streptococcus pneumoniae. This became famous for a number of reasons. A, it was the highest grossing vaccine until the COVID pandemic. This vaccine last year sold about $7 billion worth, and it's recommended in the US for anybody above 60. So most people got it, and during the pandemic it became famous because a lot of people did not die from COVID, but they actually died from Streptococcus pneumoniae, which is a lung disease. People get weak, then they get a bug, and then they die. So these three things have been used. They are isolated polysaccharides, conjugated to a carrier protein, and formulated with an adjuvant. And the idea that we had is very simple. We said, okay, we can make oligosaccharides in large quantities. We can connect that to carriers that are no longer proteins. And if you do that properly, we don't need an adjuvant. So we don't need a cold chain, and cold chain is actually 50% of the cost. So my lab has worked on a lot of different things. We synthesized and published in JAX the first carbohydrates for C. diff, a gut bacterium, in 2012, 2013. In 2015, we started a company. 2020, we sold it, and this company now, Idorsia, what bought this company, has these vaccines now in human clinical trials and are moving forward. These are very, very large markets. At the same time, my lab every year gets a new list from a WHO. This is published, and it's very simple because any of those bacteria have carbohydrates on the surface. We make them, and that is why we can actually now have programs against basically any kind of hospital-acquired infection. We are just in the process again of starting another very large company, again with a very massive investment, trying to go after these bacteria. Okay, so what I try to tell you is with automated carbohydrate assembly, you can really go after a lot of different fields, just like you can do in peptides and you can do in DNA. All right. I have to say, the way I got into flow chemistry was not what I tell you right now. The honest story is that I was really frustrated because at MIT, I had 35 people, and these people did not want to work 24/7 for some reason. So I thought, how can I make it possible that people run all the time reactions? And we started to do flow chemistry starting in 2001, because I saw a lecture by Professor Ehrfeld from IMM, and I thought, okay, that's cool, I want to try that, and this is how we got into this whole thing. But today, of course, we sell it for the reason we say, okay, the cost of manufacturing makes a big difference for genetic medicines, and the production of pharmaceuticals is very wasteful, and the output is relatively small compared to these other areas. So we've been doing this for a long time, and we make the argument that most high-value things are made continuously. Well, this is potato chips, for example, but the same thing, I think, also goes for pharmaceuticals. Pharmaceuticals are typically made in batch, and I think what we need to do is we need to actually make all these things continuously, including pharmaceuticals. So the idea of flow chemistry, and there are many of the people, the pioneers in the room here, so I don't have to tell you anything about this stuff, right? But in principle, it's relatively simple. You have different ways to put the liquid into a reactor. You have a reactor in different ways. It can be chip, coil, or packed bed, or falling film for a matter, pressure regulation, and then you collect the stuff that comes out in the bag. Funny enough, and maybe many of you remember that, when we talked about this between 2001 and 2010, people would say, flow chemistry, nobody needs it, we don't know if it's at all useful. And today you talk to people, oh yeah, we all knew for a long time, but that's the only way to do it. I think so. Finally, I think we got past that stage. What we said, okay, can we create ways to make a lot of different drugs? And what we did is, and this is again old data from 9 years ago, we said, okay, can we just take 8 different modules for oxidation, hydrolysis, epoxidation, alkylation, olefination, Michael addition, aminolysis, and hydrogenation, and can we just make a whole bunch of these drugs in here? And this is what we show in this paper, that in one set of conditions, with one set of reactors, you can go and you can make a lot of these drugs. But I wanted to talk in this lecture a little bit more about the societal applications to that. And if you wouldn't sit in this nice lecture hall here in Seoul, maybe it would be in Africa, and the best way in Africa to protect your children from mosquito bites and malaria is by using insecticide-treated bed nets. But if your kid now gets sick and has a fever, what do? You tell your husband to go down to the local pharmacy and buy some drugs, right? Now, so far so good. The problem is about 50% of the anti-malarial medication sold in Africa is fake. So the drug itself is very cheap for us. Any one of us can buy it. It's cheap. But for people there, it's too expensive. And because it's expensive, people fake them and sell total garbage. And that, I think, is really unnecessary. Because there are ways to treat this today. And I think to me it's a very interesting story. You may already know it, but that's possibly the only good outcome of a Vietnam War. During the Vietnam War, the Chinese lost more people to malaria than they lost to the bombs and bullets from the people in South Vietnam. So what they did is they started a program, and that program looked at new anti-malarial drugs. And Tu Youyou, she of course got a Nobel Prize in 2015 for the work done in the 60s and 70s by a very, very large team led by her. She looked at the old literature. She didn't look at Science or Nature or JACS. She looked at the old scrolls of the Chinese traditional medicine. And what it said there is when you take Qingao and you treat it with lukewarm water, then you have an anti-inflammatory effect. And initially they used hot water, boiling water, and that is bad because the boiling water will kill the epoxide up here. So, sorry, the endoperoxide. The endoperoxide gets killed. So if they go to lukewarm water, 70 degrees, then this moiety will remain. And through that, it took many, many years, but today about 350 million people are treated every year with these drugs. I visited the health minister of Vietnam years ago, and he said, "Sieberger, why do you work on this? Malaria is a solved problem." They used to have at least 20,000 dead in Vietnam, nobody anymore, because they make this by themselves, and not a problem. Vietnam is one of the main producers of the plants, and from the plants they extract this and then a little bit of chemistry to make this drug over here. Now this is the situation today. This is how much is being made, and this is what's needed. So why do we need more? Because population growth, global warming, so the fly is spreading, and resistance is coming in. And the promise, this delta is a problem where people make fake drugs to meet the demand, basically. So that is an issue. So why don't we just make more drug, right? It would be easy enough. The interesting part for chemists now is that this molecule is a very complex molecule, but the synthesis is pretty interesting how it's made in the plant. This is not our work. This is work by people in Hong Kong, other places. The last molecule made by a plant by enzymatic means is dihydroartemisinic acid, that molecule here, much easier. And for every kilo of this material that people extract from a plant, they get 1 kilo of that stuff over here. The interesting part, I did not know any of this stuff beforehand, is that in the plant, it's known that single oxygen gets you a peroxide. When acid is added, opens the ring, triple oxygen comes in to make again the peroxide, and that rearranges to make artemisinin. Now, that of course tells you, okay, singlet oxygen, we heard this in the previous lecture already, singlet oxygen can be made from oxygen we breathe in and photochemistry. And I have to say, I didn't know any of this stuff. My lab worked actually on photochemical conversion of oxygen to single oxygen. And again, we were the first ones to do that. This is a picture from 1912 in Bologna, where these gentlemen here did actually flow chemistry. They're interesting papers because they fantasize of cities made of glass. They're going to change the entire way of how we're going to do chemistry, and maybe 100 years later, we actually get to this point using glass reactors to make these things. So the reaction of my lab, being very simplistic, was interested in is taking triple oxygen, exciting via dyes, this one electron, get it up here, turn around, and then relax to make single oxygen. Because for us, this was the greenest oxidant to be used in the pharmaceutical industry. The problem is that it's relatively unstable, and it has to be used at the site of production. And that's why people didn't use it in the pharmaceutical industry. So we thought, okay, we could probably do that. And again, it was shown beforehand, if you are close to the lamp, the photons are there, you can do reaction. And the issue is, if you have a larger reactor, you have to basically get good mixing so that your molecules get close to the lamp. Our solution, that represents you, other people basically said, okay, can we just use a thin tubing, wrap it around a mercury lamp, for example, and then just have molecules go by there. We published this. As you're writing this other publication, we saw that other people had done this, and now it turns out one of my colleagues in Germany told me, oh, I did this in 1968. I said, how should I know? It was published in German. Okay, I speak German, that's not a problem. But also, it was never in the text anywhere. It was only a picture. How are you going to find that picture? Okay, now it turns out somebody did this also in England, I think in the 1940s already. If we continue going back, probably the Romans did this some time ago, I don't know, okay? So anyway, the idea is simple, the idea is good, and we were not the first ones to have the idea. That's all I'm trying to say. But what we did is when we actually were contacted by a company Sanofi, and the Sanofi company got $52 million from the Gates Foundation. And the Gates Foundation gave money to two companies. One was supposed to make the precursor, and the other one was supposed to do photochemistry. And Sanofi spent 25 million euros building a plant having 7 inline 3,000-liter tank reactors. And what they didn't know is that one of my students had built these reactors for other purposes, and then we wanted to just get the starting material. So I called the company in the United States. I said, can we get the starting material? They said, of course, Professor Seberg, it's not a problem. We will send it to you happily, but please assign all the intellectual property to our company. I said, I'm really sorry, but Max Planck is a federally funded research agency. We cannot give you our IP. So we went on to eBay or something, went to China, we bought a kilo for $500. So that was problem solved, okay? We got a material, we dumped it into the reactor. Early on it was mercury lamps, later we used LEDs. And what we got is basically in a shoebox, we made 150 grams per day in about 70% yield using oxygen, using a dye, TPP, and the starting material. So that worked quite nicely. So I had a meeting with Bill Gates. Bill Gates was really excited. He goes, oh, well, we love this. You know, we have a patent on this thing. I said, well, we give you a patent for free if you like it because you're going to make it accessible to poor people and not going to make any money. The Gates Foundation then gave out $10 million in grant money trying to crack my process. I didn't know why we would do that if I give it for free, but anyway, don't have to understand everything. So we said, okay, this is good, but we got to get better. And the big problem we had was that we had to use this dye, because the dye is A, expensive, and B, we have to separate it after reaction. So we said, okay, that's weird, because in principle, everything should be in the plant, everything that we need to do here. So I hate gardening, I admit that, I don't like green stuff, but that's different to my problem, right? So I had to think about plants, and we had to think about the fact that everything should be there. And if you like plants, and if you like chemistry, it wouldn't take you as long as it took me. But in principle, this is chlorophyll. And chlorophyll and TPP, for all intents and purposes, look exactly the same. So we said, okay, now what are we going to do here? We're going to do really green chemistry. We're actually going to take a green solution. We take Artemisia annua, we dump that into toluene, take the whole thing, add acid, pump it through, and make artemisinin. And that actually works. You can also take lettuce or anything else for that matter. We tried that as well, but you don't need to add TPP anymore. In principle, we mimic the natural process. In 15 minutes, this whole thing is over. So that is very, very nicely scalable. And then what we wanted to do, we actually wanted to use it to make a lot of malaria drug. The problem is, typically this is produced in Vietnam or southern China. It's extracted under horrific conditions. And then they do 2 steps of chemistry, and then they sell it afterwards. And then the big pharmaceutical companies fact it up by a factor of 14, and then they sell it. So what did we do? We said, okay, let's just start a company in the United States, in Kentucky. If you know Kentucky, what do you know about Kentucky? There's bourbon, there's horses, and there's tobacco. And tobacco fields, we don't need so much anymore because people smoke less. So we said, okay, turns out artemisinin grows the same way. You grow it first in a greenhouse, you plant it, when it grows, and by the end of the day, it's about 2 meters high. It's about as tall as me. So we actually grow large fields in our company in Kentucky. And what we then do is we start looking at this and trying to use it as a malaria drug. The interesting part, for me, I'm originally a biochemist, is that artemisinin is not supposed to only work against malaria, it's supposed to work against pretty much everything else. And to me, that made absolutely no sense. Why would you have a drug that works against everything else? Well, also against cancer. Professor Effert in Mainz, German guy, he reported in 2001 it should work against 115 different types of cancer cell lines. I thought that's all bullshit. I thought that can't be true. So what we did then is we did a lot of work on a mechanism. I'm not going to talk about it here. We know now the target. We know what it goes after. And people at the University of Kentucky Marquis Cancer Center got so excited. This is actually the annual report a few years ago from them. This is one of our co-workers here who grows this stuff. We went into a phase 1 dose escalation trial of advanced ovarian cancer in females. When females get ovarian cancer, when typically in the United States they get surgery, and then about 80 to 85% have recurrence within 18 months. And what we did in this case, and this sounds totally idiotic, we took teas made out of special Artemisia annua, which we grow in the United States. It has very, very high content of active ingredient. And in this case, we had less than 5% of the females had recurrence. This was only a phase 1 trial, so we only went for toxicity. We did not go for treatment. But the females didn't want to come off the treatment. So to this day, we are staying on this treatment, and we had very, very few females with recurrence. The hospital got so excited, they said, okay, we would like to go in a phase 2 trial for prostate cancer. And today, already for over a year now, we have this material in men with prostate cancer. Why did we switch from females to males? Not because we are sexist, okay? No, because in prostate we had advantages. There's a PSA level. So a prostate-specific antigen. You can measure that, so you can actually get a measure, which in ovarian cancer you don't have that. And what we are seeing now, I can't tell you the results yet, but we are extremely optimistic because we're actually seeing the PSA levels of the people that are enrolled in our trial. And right now we're getting ready to start another 12 trials in other cancers at this stage. So actually this stuff is pretty interesting. From a flow chemistry standpoint, of course, we wanted to say, okay, can we maybe marry the way of synthesis in flow, but in a synthesizer we do not have to constantly change matters. And what we showed in this paper here is basically by arranging the different types of pumps and reactors and analysis around a central switching station, we can just adapt different types of chemistries into the same reactor without having to change the whole setup. If you look at your company, I just saw that you're also using this flow reactor. This is now the reactor before actually our lines are put in place, otherwise you don't see anything afterwards anymore. This is the size for an American refrigerator basically. We have pumping systems, you have mixing systems, you have analysis systems, and you can go through different types of chemistries up here. What we showed afterwards also is we made very simple compounds such as paracetamol, but also these anesthetics. Why did we make these compounds? During the pandemic, in North America, Canada, and in Europe, these drugs were actually not available anymore because they're made in Asia and they didn't get shipped. And so people came to us and said, well, Peter, can you make these drugs on site? We did that in flow systems, we tried it out, and then we scaled the flow systems to large scale to actually make the API for human use. And in my title, I think democratizing production is actually exactly that, because by the end of the day, I would like to be able to create production sites, maybe not in the remote islands, what was described by Volker Hessel earlier, but maybe in different countries that don't have pharmaceutical production. That's our goal, basically, and building these types of systems for people. So the goal is really to have this chem farm concept where, like a server bank today, we can have standardized reactions where you basically have your different kind of chemistries at remote sites that anybody can access. Now that seemed maybe like a total crazy dream, something that maybe somebody could do, but the other thing we knew is this would take a lot of money. And that gets me biographically to the last step, which started about 3 years ago. The German government, Chancellor Merkel at that time, and one governor, they decided to take 2.5 billion in total, and they would have a call for proposals to fund 2 research centers. And I didn't want to do that, but I was asked multiple times by ministry to apply. And I told you what I usually work on is biomolecular stuff and vaccines and things like that. But I realized if we do this, we're going to take a different topic that has to do with flow chemistry, or has to do with engineering, but we should go after a different and more comprehensive approach. And really what I go after is we would like to create a sustainable circular economy. This is a basic funding for the next 15 years. Afterwards will be more funding, and the goal is to have 1,000 employees. So the idea is very, very simple. Today we have basically a chemical industry that's built on gas and oil, and I should say the idea was already put to the government before the Ukraine crisis. Of course, ever since, it's become a lot worse in Germany. We are running low on gas and oil. Raw materials are made, products are made, waste is being produced in addition to products, and the issue is CO2, wastewater, and energy. I think what we can do relatively quickly is that we're going to reduce the amount of fossil fuels that go in. We can begin to think a little bit about becoming circular, but if you want to go a radical step and saying we really want a circular economy, but we're going to go with raw materials to products and bring this really all the way around, that is a gigantic task that's going to take all the chemists around the world, I think, the next 30 to 50 years. And we felt that if we get significant money from the government, we should really think about very broadly about chemistry, catalysis, engineering, waste valorization, energy sources, but also new product design. So a very comprehensive approach. So what we did then is we identified 5 thematic areas that we were going after. One is the area of automation and standardization, because I think we need to get good data in order to be able to do really data-driven chemistry, as we call it now, or AI and chemistry, depending how you look at it, because the literature, I think, is full of garbage data, and if you take garbage data, then you're also gonna get garbage conclusions afterwards. You're gonna think about recycling, we're gonna think about renewable feedstocks, but also we're gonna think about society, because society is gonna be willing to take up this new chemistry, which may mean new products, but it also may mean different types of prices, okay, in the future. I'm not going to go into details now. This project has gone on now for 2 years. We're actually building the center today. We had about 45 people. I still have quite a ways to go to get to 1,000, but we're getting there now. And we identified 2 so-called moonshot projects. One of them is to make a regionally sourced, 100% recyclable car, not using any fossil resources. Now this came up because I sat with the head of a board of BMW, and he said in 2040 BMW would like to make exactly that kind of car. That's only 16 years away. And he said, okay, that sounds pretty crazy, because you think about how complex a car is, it's all chemistry, right? I mean, most of the things are now plastic, it's glued together, you need lubricants, I mean, think about a battery, the sealants, the whole thing, it's just chemistry everywhere. Germany is a car nation. I think Korea also lots of cars, but I think Germany, car industry is very important to us. And we believe we can discover new pipelines here, but also new processes and material. So that's one of the 2 projects where we are moving forward. The second one I'll talk in Busan a little bit about is a totally automated lab. I would like to create labs that have no longer humans in them. I think a lot of chemists really worry about this, but I think that's really what we need, because Germany is so expensive. I think Korea getting there as well. We pay people a lot of money, and the only way we're going to be able to compete against low-wage countries is if we're going to automate as much as possible. I know it makes chemists nervous because they think we're going to replace all of us. I think that's nonsense. The opposite is true. If you automate, more chemistry will be done in these high-priced countries. I show this for a simple reason. This was at the time we submitted this grant, and Volker Hessel was on the panel. He saw it. We had 140 companies. Today we have 200 companies involved with us. Some of them you may suspect, people like BASF, who leave Japan and go to China, I guess. Bayer and the large ones, I think those would be a given one. What's interesting, we have one of the big supporters, BMW, but also Microsoft is one of our major supporters, because they are really interested, not just, the other thing, these are, you may have never heard of them, UPM, Mercer, and Stora Enso. These are 3 of the largest forest companies. Because what happens today is when you take wood and you make paper, you burn 50% of the biomass. And from that biomass, you can actually make very interesting plastics. So that's one of the sources of how you can start to think about making recycled materials. So all these companies, the unions have come together to do all that. What we create also is university network, local network around Leipzig, regional in Germany with TU Munich and others, and globally we had MIT, my former colleagues, Oxford, Riken, and Glasgow. And at this moment, the only thing I can say is CTC is new. CTC is going to try to reach out globally, and I'm completely convinced the only way we can transform chemistry into what we think should be done is if we do it all around the world, if we do it together, because the task is really humongous, and nobody, no center of 1,000 people can ever do this by themselves. I think we need to do this together. With that, I will just say, I think chemicals are a key industry. In Germany alone, it's the 3rd largest sector, 190 billion in sales. 97% of all products sold in Germany are made chemically or treated chemically. We gotta change it, and I think the way we're gonna do it is shown here. The transformation is gonna come. Let's do it together. CTC is gonna be part of it. And with that, I'd like to thank the people who work with me directly. My own group is about 100 people at Max Planck, but that includes different assistant professors. These are the people who work directly with me. I think 17 different nations at this moment. Actually, no Koreans at that time. Some Japanese, some Taiwanese, but that, of course, has changed over years. I thank the funding agencies, mainly from Germany or the US. These are the electronic means for you to get in touch with me. But since I'm here and you are here, maybe just ask a question. Thank you.
Amazing. So my heart is beating fast. Because it's a German, it's somebody like maybe 1 century ago. I think that's the kind of origin of some organic chemistry. now this is a government like a drive, like a new program for the sea water. If I'm young and then I'd like to join this one. Okay, question. Please.
Thank you. I think the Chem farm concept is very innovative, but the transformation of based on current situation, the transformation of petrol refinery to the bio refinery. What's the most difficult problems for the transformation?
I think it's a total misconception, and I think I paid into that as well, that we can go to completely bio-based systems. This is totally impossible. If you look at the amount of carbon that we use today from petrochemicals, we cannot substitute that with bio-based. It's not possible. The amount is simply not possible. So recycling is going to be an important thing, and ultimately it's my belief, looking at the numbers now, the only way we can do it is if we actually utilize CO2. So I think what we really need is ways to actually get to the CO2 and then do catalytic reactions using CO2 and making base materials. Because if we really go to a system and say we don't use petrochemicals anymore, our limiting factor is going to be carbon by the end of the day. And if you look at the numbers, people talk about this all the time, right? Oh, we can use algae, I mean, you're going to use wood. I showed that too, right? But compared to the numbers we have today, it is very, very small. So we need other ways to go to carbon. And recycling is one possibility. Also, with what we have today, the plastics we have today, you can't recycle most of it, right? Forget about it. And I think in that case, the best way would be to use CO2, which is a difficult task.
Okay, actually, out of your talk, and that is something that you try to use a polysaccharide as a kind of some of a specific adsorption for the target drug delivery, and by linking with some carriers, right? So, and there is some of the polysaccharide is as a some of like a specific adsorption. So, what kind of advantage compared to the some peptide or antibody or the other?
Have polysaccharides? I think polysaccharides, for the most part, are not immunogenic. So if you look at peptides, many peptides are immunogenic if you use them as drug delivery vehicles, for example. So carbohydrates are actually very benign. If you think about the COVID vaccine, I don't know in Korea, but in Germany and I think in Korea the same thing. One of the major issues they had is that they used PEG chains on the outside of the vesicles, and there are known immunological reactions to PEG chains. And the PEG chains are used because in principle they are sort of mimics of carbohydrates. So if you use carbohydrates such as mannoses, which are also on HIV virus, by the way, they are totally immunosilent, so nothing will happen. And I think that's one of the advantages of carbohydrates. The other advantage, of course, is eventually it can get broken down in the body and nothing is left but glucose or galactose, and that's not really bad for you. So I think that's one of the things that people can think about. The problem of carbohydrate has been that it's difficult to get to the carbohydrates in pure form. And I think that's one of the limitations. But I personally believe a lot of the things we're going to see is from seaweed, from algae. I think in Asia, in Europe, you tell people to eat algae, that's usually not very well received. I think in Asia, most places, it's absolutely perfectly acceptable, right? I think in China, there is one drug that people believe works against Alzheimer. Based on mannose. I don't understand how it works, okay, I can tell this much. But I think there are a lot of interesting things that could come from the ocean, because the problem is, I showed you the synthesis, but the synthesis that we do is done on small scale. So if you need for a vaccine, we need, 5 micrograms. The cost of goods is irrelevant. But if you really want to make a drug delivery molecule and use our method today, our method is too expensive. I mean, eventually it can probably be scaled, but I think if you have a way to make something from nature, you'll be definitely cheaper by the end of the day. Interestingly, just as an aside, there was a blockbuster drug carbohydrate, it was called glucobi. It was an isolated tetrasaccharide. It worked really well against diabetes. The problem was it was socially unacceptable because people taking the drug got huge gas production in the intestinal system. They never took the drug anymore, but otherwise carbohydrates can be good drugs.
Thank you. Yes, please.
I was so impressed about the CTC project. It was really, really nice. But I would like to ask the question about out-listening. I was really surprised even out-listening containing T is effective. And that means I think that orally available, out-listening is already available. Is it right?
Yeah.
And out-listening is the once the human that you take that, in the gut system and the stomach system,
Yeah.
it is stable.
Yeah.
That peroxide moiety is not creeped and just going to the stream.
Yeah.
Oh.
So I was totally surprised. So I can tell you when I first saw this, right, I thought, okay, that sounds like total nonsense. I mean, you take a tea, that's totally crazy. It's not gonna work. There's a woman in the United States, Pam Weber. She's mid-70s or something. She's worked on this for like 25, 30 years. And she has done a lot of this uptake stuff, not all published, but talking to her, they get very high blood levels. The issue is the wild type sweet wormwood in China has 0.1% of API. In the ones we grow in Kentucky, these are specifically grown plants we developed. We have 1.8%, so you have 18 times more. If you would take the stuff in Germany, for example, during the pandemic, they sold this to everybody, and there's nothing in there. Okay, you take your money, throw it out of the street, same effect. Interesting. So I took this during the pandemic. This works against COVID. There's now a phase 3 clinical trial was run in Madagascar, where people tried that, and they had extremely good effect. We know now why it works. My lab discovered the mechanism. The mechanism is where they say palmitoylation gets blocked by artemisinin. And when the spike protein has to trimerize to attack in COVID, it has to be palmitoylated. If it's not palmitoylated, it doesn't attack, it doesn't trimerize. So actually this molecule blocks the palmitoylation, no trimerization, no attack, and that's why it works against so many viruses. Herpes virus, all other things. In Germany, it's not allowed against any of these things. In Germany, the sale of these teas is not allowed, it's illegal. Some people apparently sell it, and I don't promote this, but I understand biochemically now how this works. And 5 years ago, I thought this is all complete nonsense. And we also know now why it works against cancer, because in the RAS pathway, there's also certain palmitoylations which take place in signaling. And I think that is what this thing basically modulates. But I can tell you many people, including myself, that are usually working on drug discovery, when you tell them, oh, you take a tea, there's lots and lots of, I was totally skeptical, okay? But I think today what we see in these people in the United States, in totally clean clinical trials in the US, very, very clear effects.
I think that, please let me confirm that the tea is effective for cancer prevention, right? Is it effective for the parasite, malaria?
So that's one of the reasons why people don't want you to use it. So the reason is that there is resistance against artemisinin setting in Laos and in some parts of Africa. And so the problem is what they do is they take pills which have more than one drug. It's not just artemisinin, it's something else. But artemisinin, for example, works against other parasites as well. But the WHO has been very, very strict that you shouldn't take a monotherapy, okay? And that's why I think people in Africa shouldn't take it all the time, because eventually a parasite is going around with it. For cancer, it's a different story, right? I have to say, we have not proven that this is preventative. We are right now, the company is getting ready for prospective trials. We're going to take a large population, give it to a large population, follow them over the next few years, and see if it can be prevented. A lot of people believe that. I'm a scientist, I think let's do experiment, see what happens. But of course, a preventative trial is much more involved because you need a much larger number of people. You got to go out and do a lot more of that. I think it's interesting now in the US because with Trump, it could be interesting because there's now all sort of esoteric stuff coming into play. We'll see what happens. But one of the reasons why Kentucky did that, Kentucky in parts is a very, very poor state in the United States, and they're underserved actually on the cancer end of things. And the hospital paid for most of those trials, so our company didn't pay for it. Actually, this was paid by the hospital because the hospital has a real need trying to come with less expensive medications.
Thank you so much. Yeah, I could understand.
Thank you. Thank you very much. Very amazing talk. I'm very amazing about the CTC program and also amazing about the polysaccharide synthesis by automation. So what is the range about polysaccharide polymer produced by the automation? That is one question. And second is how many polymer can use as a vaccine? Yeah.
So we have made up to 100 mers, 150 mers, but I don't think that's what you want and need. For vaccines, the typical antigen size that's effective is between a tetrasaccharide, 4, and the longest ones which we have in trials, I think, are 15 mers, 16 mers. So that's sort of a range which is not difficult. There's one associate professor in my department, she works on material science, and her students, undergrad students, they make every day 120 mer. So a 20 mer, because we get about 1 per hour, so in one day we can make a 20 mer, and they just make small modifications like in peptide chemistry, and they look at aggregation. They are building sort of carbohydrate origami now, which is totally useless, but just cool to do. And actually, I have one Korean student who was one of the big developers in my lab. He had 18 papers with me, one in Nature Communications, and he is back in Korea now, and he was one who really used to synthesize this. I'm not exactly sure where he's working nowadays because he was in the US for a while, tried to become professor in the US, and then eventually came back to Korea. But if anybody in Korea needs to make it, I recommend that guy. He was one of the key developers in my lab about 10 years ago in this technology. I think the issue is really, as I said earlier, the scale is relatively small. So we typically make 100 milligrams to gram quantities. We could probably scale up to kilos, but above that, it would not be doable.
And does it polysaccharide vaccine can be at anti-cancer vaccine or yeah?
So I started my postdoc at Sloan Kettering Cancer Center in New York in 1995. At that time, Danishevsky's group looked at anti-cancer vaccines because there are specific things in breast cancer in women, in prostate cancer in men. Sorry, it's a little long-winded answer, but they looked at global age, it's hexasaccharide. They made it in 95. They moved it into a company by Chi Wei-Wong into Taiwan because in Taiwan there is a very, very large incidence of breast cancer in women. And they ran up to phase 3 clinical trial, phase 3 human clinical trials, and it failed. And I saw that, I looked at the clinical data, and the interesting thing is 50% of the females responded. Wow. And only 50% of the females met antibodies, because the problem is when I give you, any one of us, a carbohydrate, it's a very poor immune response. Oh. So if 50% of females that responded had antibodies, they got rid of breast cancer. But because we ran the clinical trial wrong, the company failed. So I looked at the data because I followed this for 20 years, right? I saw this, and immediately, 2 years later, we started a company in Germany, and we are not doing active vaccination. We are making actually monoclonal antibodies and nanobodies, because when you do passive immunization, so you make the antibody, and then somebody gets sick, then you're going to give them the antibody or an antibody drug conjugate. So my company, Takalix, sorry, it's not advertising now, right? But our company is basically making the antibodies, but it was really, really difficult to make good antibodies against carbohydrates, because carbohydrates are not great immunogens. We ended up having to screen everything from chickens all the way to alpacas. My group leaders also wanted sharks, because sharks, but that's when I stopped it. Okay, sharks, and they're not going to do it. And finally, we were able, but it took us 5 years to get this going. But the approach is not active vaccination, it's passive vaccination, so basically giving antibodies. That's how we go after it. And the other reason is, I'm not really sure it's a good thing to vaccinate people against human structures. Because if you think about it, the carbohydrates on cancer are self structures. And if you immunize against self, you make it autoimmune disease. That's why I always focus on bacteria and parasites, because they are very different. Whereas if you go to human systems, mammalian systems, I think it can become a little bit tricky. So that's why I always stayed away from this.
Thank you. Thank you very much.
Okay. Thank you very much. Actually, we have a schedule, and then it's some the panel and discussion, but the 5:40, and then but already every talk, and then individually, and then separately we have enough discussions, right? So it's a normal talk, but we have about some 5 minutes before some the group photo or take a pictures, right? That is a kind of some the official ceremony, right? But and then I'd like to ask you, if you don't mind, and there is a circulated some microphone, because it's 4 speakers, and there is some already they have a chance to introduce, right? But and then is some 3 speakers, and then it's in the Busan, and then have not have a chance to introduce yourself. So then and then it's another audience. And then I really appreciate it. You can keep the position in the seat by the end of some of these symposiums, right? So it's please some if you don't mind, and then you can introduce, right? Do you have so many microphone? Okay. 이거 무선이 아니네. 무선. 거기 저기 후세 선생님.
Okay, my name is Shin Ichiro Huse, but my first name is too long, so just call me Shin. Yeah. In Japan, the Shin means new. Yeah. And I came from the Nagoya University, and I really love the microflow synthesis. I'm a chemist, and in particular, I really now doing the peptide synthesis. I think that I can really introduce about my peptide synthesis in the microflow reactor. And I really like the bio-oxidation, continuous bio-oxidation in my body. Flow chemistry. Yeah. Drink, ethanol, aldehyde, acetic acid. Yeah, I really like the flow chemistry. So that from tonight, I think that I really expect that we can enjoy so much. About the bio-oxidation. Okay. Yeah. Thank you so much. Yeah.
Thank you very much.
Yeah.
Hi, my name is Michael Van Dam. I'm a professor at UCLA in the Department of Pharmacology. My background is in kind of engineering mostly, but I have come to work on microfluidics for synthesis of radiopharmaceuticals. And these have interesting challenges due to the very short half-lives and the very small quantities we need per batch, essentially. So I'll be talking about some of the technologies and techniques for discovery and production of those.
오늘 안녕하세요. Hello everybody. I'm Hirotsugu Usutani from Sumitomo Pharma, and I'm a process chemist at my pharmaceutical company. And when I was a student, I studied at Professor Yoshida's lab. So one of my purpose at my company is applying flash chemistry to process development. So I will talk about such a kind of details in Busan. Thanks for today.
My name is Liulan Shen from Smart Biopharm in Korea. Smart Biopharm is a venture firm. Yeah, focus on the flow chemistry process development. I usually develop the API process or intermediate process. Recently, I wanted to develop the process to business. So tomorrow I went to China for my business trip. Yeah, so it's very sorry that I cannot to go to Busan. So it's very, very, very sorry to all of us. Yeah. From Kyunghee University. Yeah, in Korea. Yeah. And this is my secret that I will naturalize to Korea. Yeah, immediately. Yeah. Thank you very much. Yeah.
Okay, my name is Changjin Lee, and I'm working in Korea Research Institute of Chemical Technology. As I mean that I'm interested in the chemical technology, and I'm mostly working on the polymers. So I'm working a few experiments using the flow chemistry, making a polymer, and some are successful, some are not. Okay, and I'm not the registered member for the Busan symposium, but I'll attend it anyway. Okay, and I will see it in the Busan. Okay, thank you very much. Yeah.
Nice to see you all, and I'm Sang-kyung Kim from Korea Institute of Science and Technology, and I am working on let's say in vitro diagnostic systems and PCR systems. It's a little different from like major interest in this area, but my major in college was chemistry, so I was trying to follow this chemistry today, but it wasn't easy. It's a while since I've seen these chemical structures and everything, but still I'm interested in functional chemical materials, like fertilizing the diagnostic capabilities, like encapsulation, and try to trigger some like action point. So still I'm trying to follow you guys, and then I'm learning a lot. I'm expecting a lot too. Thank you.
I work at the Korean Association of Senior Scientists and Engineers. One member of our association is established the International Vaccine Research Center located in Seoul National University. I really very grateful for all of today's speakers. Some space chemistry, flow chemistry are very interesting to me, and I think that forward regions in chemistry. Thank you.
저쪽에 또 괜찮으시면 소개하시죠. 네. 친구들이 더 오니까.
My name is Pilgu Lee. I'm running a venture company, pharmaceutical venture company. Then I am medical expert. I'm medical doctor, also being involved more than 25 years in pharmaceutical industry. I just learned to what is flow chemistry to adopt manufacturing, because we are one of our pipelines is liposomes. So probably I will questions in Busan. Yes, I will do. So, yep, I will talk more about our pipelines next 2 to 3 days. All right.
This is John, Kenny. Actually, I'd like to introduce one guy. Actually, he's comes from my group, only part of them. But then actually, I have to like appreciate it. That he is Kisuna, and then actually he really organizing, and then he communicating, and then it's without his assistant, I think that we cannot make it. Yeah. Also, and then the cost some. The money sponsoring is also important too, but he make it a lot of work, right? Okay, so also Amir he's from the Iran, and he's on the PhD program in my group. Also, and there is Sanjeev. Yeah, Sanjeev. Your last name is Sanjeev Singh. Kumar. So, he's postdoctor in the organic chemistry background in my group, right? So we are very, interdisciplinary. So it's Busan, and then probably the other members are going to join us. So is there any other members that want to introduce you? Steph? Yeah, I think that this is Steph from the coast, right? Also, it's a thank you for your help, right? Thank you.